Hi there. Thank you for tuning in. What follows the 3-hour session of the MYC.NYC online classes launch that was recorded on Friday, March 27th, 2020. Improving scientific literacy through the lens of mycology and soil biology is a pivotal step towards participating in citizen science and making impactful positive change in our world. Through diligent observation of our surroundings and intentional application of beneficial microorganisms, bioremediation, carbon sequestration, and regenerative agriculture can be made a reality through our combined enthusiasm and effort. If you enjoy the content that I produce and appreciate the work I put into it, please consider supporting me directly through a donation that you feel is appropriate. Links to donate can be found on my Instagram through my link tree. You can donate via Venmo, PayPal, or through LiberaPay, an open source Patreon alternative for recurring donations. Thank you. We're going to get started. So we'll basically go for roughly, 45-ish minutes, and then we'll answer some questions. I'll try to consolidate the ones that overlap. But yeah, here we go. Cool. So yeah, so who am I? My name is Craig Trester. I'm a educator and citizen scientist. So I kind of got into mycology, mycelium, mushrooms, the whole world of fungi back in 2016. It was kind of where I was an interesting kind of transitionary period of my life, kind of going between careers. I was working a service job, so I had some flexibility, kind of asked myself what I wanted to do with the rest of my life. And I was interested in looking into like solutions to the ecology environment, because I had studied environmental policy and during university. Now, it was kind of interesting because I was reading about bioremediation, about phyto and phycoremediation plants and microorganisms, but then I heard about micromediation. Probably we've all stumbled across like the Paul Stamets talk, like 6 ways that mushrooms can save the world, and it was kind of interesting because I was like, whoa, mushrooms can do that? Like they can break down volatile organic compounds, and I was thinking, why do I know so little about mushrooms? And it was kind of crazy. Like I kind of had this mental flashback to my intro college biology lab. We got handed a course packet, and then in it we had a bunch of pages that overview the different domains and kingdoms of life. And it was, every kingdom had a page and a half, and then fungi had a paragraph. It was like, decomposes organic matter, makes relationships with trees through mycorrhizae. And it was like weird. It was like, okay, why do I know so little? So that was kind of this whole thing where I went straight down like this rabbit hole, kind of looking online what I could search and learn about, and which ultimately led me to joining the New York Mycological Society. And the New York Mycological Society is probably one of the older ones in the United States after the Boston Mycological Society. It got founded in the 19th century and then kind of had a resurgence with John Cage, the minimalist composer, back in the 1960s. So here I am with, from right to left, Tom Bigelow, he's the president. Sigrid Jacobs, who's one of my really good friends, she's awesome, and myself. And I was presenting at this event called Understory back in, I think it was 2018, where I've actually probably met some of y'all. But it was really interesting because I was like, how do I learn more about mushrooms and fungi? So I joined this mycological society or mushroom club, and it was cool just going out and exploring the world and environment, learning about like my ecology. And it's tough in an urban environment. You're like, okay, I have a couple parks, and it's interesting. You think about how we tend to have our perception about iconography or visual our visual acumen is more tuned towards things that we constructed, artificial things. Like, the average person can probably recognize over 200 to 300 different brands of companies or corporations, but probably less than 5 native plants. Even fewer mushrooms. But it was amazing because it kind of engaged me in nature in a way where I, rather than just being in a space or hiking or camping and being, oh yeah, that's that green stuff or rocks or whatever, I could start to appreciate it more. So the New York Mycological Society really kind of dove me into that, and I started getting really into how do I apply and work with it, and hearing about micromediation. Went to a workshop that was hosted by 2 ecological program managers up in Vancouver Park, which was mind-blowing and awesome. And it was a mushroom cultivation micromediation. That's when I learned about mycelium running, radical mycology, the mushroom cultivator, all these books, and kind of, eventually I started to basically, have basically do this lab work at home in my kitchen. Roommates threw it out a couple times, really confused why I was growing mold on little plastic petri dishes in my refrigerator, and kind of evolved from that different capacity as well. But yeah, so eventually, kind of after going on lots of mushroom walk with these amazing folks, I eventually kind of stumbled upon the kind of community biology scene in New York City. So there's two amazing labs in New York called Biotech Without Borders and GenSpace, community biology labs where people come want to learn who, maybe not in an academic setting that are curious about life sciences, but beyond an aspect of theory, rather than just reading a textbook, but physically doing and acting. So it was an amazing kind of combination of DIY, hacker space with applied biology. So I was really interested in bioremediation. So I was interested in applying for the space of doing a micromediation project. And I was like, okay, cool. Yeah, I just want to experiment and try around and try things out. And then pretty much they asked me if I wanted to open source or open up my project to the community at large. I have a micromediation project. I was like, yeah, sure, that sounds cool. Maybe maybe 5 people will join. 3 people will fall off, me and 2 other people messing around. People started showing up like in the groups of like 10, 20, or 30 sometimes, and it ended up kind of throwing me in this teaching environment all of a sudden. So it's really kind of interesting to see this. It's a kind of progression. But the one interesting thing about the community biology scene there was that it has a background or basis in molecular biology. So really kind of understanding that genetic information is the means by which life evolves and stores information and progresses and basically helps it survive by expressing these genes. And it was amazing because at the same time I was just looking for a space just to grow mycelium out on petri dishes and experiment and just learn more by doing in general in this experiential capacity. I kind of dove into learning molecular biology. It was actually a prerequisite for joining the lab space. So it's amazing. We're kind of realizing now how at this point we're realizing how important molecular biology is, and even to the point where kind of hand in hand, we're in the 1969 fungi were made separate from plants in an official kind of taxonomy standpoint. But it was the rise of molecular biology and DNA sequencing, it becoming affordable in the late 80s, early 90s, that fungi, we could clearly say they were separate. So it was amazing. At the same time, mycology kind of came in its own form, so did molecular biology. So it's this amazing concept in time in general. So it was kind of serendipitous that I happened to stumble on 2 spaces that allowed this aspect. So I kind of got into DNA barcoding. So the New York Mycological Society is one of many upon many mushroom clubs and organization societies around the country, but also the world. And there was this event called the North American Mycoflora Project. So it's a project where people were amateur mycologists, which the New York Mycological Society is, as much as some people are incredible in working out a taxonomic key and understanding the environment and ecology of fungi, partnering with professional mycologists to collect specimens and samples, extract DNA from tissue, amplify and sequence it. So kind of having this amazing like molecular naturalism to kind of explore and express. So it was something that was really incredible and amazing, that opportunity. So I kind of dove into the Mycoflora project. I was working a service job as a bartender and barista, and had all this free time. So it was kind of amazing to look back at it now. Yeah, so at that point, I was doing it and people were interested in and what's really interesting is that working with genetic information organisms, oftentimes the protocols you're working with are a bit complex. If you're trying to isolate DNA from mammals, you tend to have to take interesting precautions because oftentimes we're a vector of contamination for our own genetic information. You have to wear gloves and a face shield and other stuff in general as well. Working with plants, a little more complex, and you often have to kind of get access to the chloroplast. But with fungi are kind of amazing because they're these polymorphic structures where their tissue is kind of like a mix between like a stem cell and a nervous cell, and they're packed full of nuclei. So using these different regions you can barcode them rapidly. So that was kind of a progression from teaching into doing and teaching into doing again as well. Eventually people started asking me if I wanted to teach. I had friends that ran mushroom farms, taught DNA barcoding workshops in the field. That was really kind of crazy, having a lab in a bag going through security in an airport. Here's a kind of compilation of workshop I taught at my friend's farm, Micro Uprisal in Tenino, Washington, just outside of Olympia. And also, too, apparently someone came and did a profile on GenSpace, and it was really interesting. And this photographer showed up. So, what are you working with? Oh, I'm working with Petri dish. And it turns out that the story was, it was interesting. It was kind of more like this cautionary tale about, you know, community biology spaces, how they're going to make a new virus or all this kind of thing. And, or even the reality in general is it's really hard to try to do that. It takes a whole team of PhD students, postdocs, and even people that have been in the field for decades to kind of assemble a genome in that capacity. So it was funny, like this big scary story about community biology, and here I'm holding a petri dish with a lot of bacterial contamination on it when I was still learning, and kind of ended up on the front of the Science Times, which was interesting. I've been reading it since I was a kid, and the person who ran the lab freaked out. I was like, oh my god, I'm sorry, it's cool, man. Like I'm not working with I was back when I was just culturing in general, and apparently now I'm also on the New York Times app under science, so kind of crazy that capacity as well. Anyway, yeah, so eventually I kept on teaching at community biology spaces. I had the opportunity to teach, you know, DNA barcoding, which led into teaching general mycology, and then people asked me if I wanted to like teach a class, and ended up with admissions, and then eventually I had the opportunity to work with an amazing educator, Calvin over here, who I'm circling with my mouse cursor. He is the director of youth programming for Beam Center. Beam Center is a makerspace for teens based in Red Oak and Brooklyn. And so they're all into experiential education. And he approached me to take some classes, a tissue culture class, and then a general lecture, and said, "Hey, you know, I work for this organization. We do these apprenticeships around experiential education, a hands-on education, really kind of making and doing, which engages the tactile kinesthetic kind of spatial intelligence, where often a lot of curriculums focus more on the reading, writing, verbal, and mathematical intelligence." So it was an amazing opportunity to design the 16-week curriculum around fungi. It was called Fractomycology, an amazing group of students making and doing built equipment as well. It was an amazing process to introduce them to life sciences and also biology, but this whole kind of world where oftentimes it's unapproachable or seems inaccessible. You know, oftentimes it's not visually presented. So it's kind of amazing. I like to think about fungi. There's this kind of weird non sequitur where people know so little about them, and it's such a thing that catches them off guard. All these preconditions about what it means to them just don't exist, or just kind of are taken off guard. So it's a nice way to kind of get people excited about education and biology. Went out, basically did the whole tour. This is in front of Smallholder, a mushroom farm based in New York City. That's my whole group of students in class, and my really good friends, Louis and Adam, that actually that run Smallhold as well. So it's a great example about the emerging the bioeconomy. How, you know, there are careers in biotech and biology, but these subdisciplines of sciences of mushrooms are kind of this huge economic opportunity to, you know, but also kind of lift people up to learn about natural environment, but also even do things with them as well. So it's amazing how big the potential is for mycology overall. Here I am doing a BioBlitz with Macaulay's Honor College as a taxon leader. So we're looking for mushrooms in Greenwood Cemetery and over by the Gowanus Canal. So showing in even urban environments, life is here and present in doing things. And, you know, we're kind of decoupling this very anthrocentric kind of view of the world that we have as a human species. So there's kind of some mushrooms we found in BioBlitz. General introduction. Yep, you're laying out again because I had to. Even to the carryover to art school. So you're hearing about School of Visual Arts with Suzanne Ankler, who's the BFA chair, kind of the person that more or less like straight up coined bioart and kind of really kind of laid the book down on it. But talking about the potential for like what fungi mean, not just for like a scientific capacity, but for mycomaterials, for biological art. Now, it's really interesting about something usually we think about art as a stable concept, but you know, it's interesting because fungi have this ephemeral quality to them. They're decomposers and have all these interesting, deeper connotations we can kind of extrapolate and look upon. So that was a really kind of interesting aspect overall. CUNY Baruch as well. I'm a guest of my friend Helene Eisman. She's a instructor over there as well. So kind of the fact that due to this vacuum of knowledge around fungi, I had a lot of opportunities to teach and really tried to make it accessible as possible, given the fact that, you know, I grew up in New York City as a product of the public school system, and really, I felt that's a big part of what I do, giving back and creating opportunities and really kind of not having this transactional approach to it, but really kind of free and open to kind of make all these possibilities happen. Even had the opportunity at Biotech Without Borders, which Community Biology Lab, to manage the lab for the summer for their program called iGEM. iGEM is a International Genetically Engineered Machine competition. It's a competition that tries to use molecular biology to develop solutions for many of the world's problems. So those are two of my students from Beam Center actually were brought along with us, Josiah and Zinwei. And then two of my interns, Arietta and Kevin, who are really awesome, the two collegiate solid interns, and really kind of integration of molecular biology, but also to deeper kind of ecology through fungi, and kind of looking at these interesting interchanges of how doing actual work that requires like discipline and practice and working with a pipette is an interesting experience and practicality, but also a lot of the DIY low tech examples of working with mushrooms and kind of getting engaged in the environment. Here am I teaching at NYU, teaching my friends Oko Farms, which is a aquaponics farm. But yeah, that's basically an introduction as well. So we'll kind of get started now with the actual meat and potatoes of the presentation. So yeah, and feel free during the session to ask questions. You can see the question link up, they'll populate, and we'll probably stop around maybe, probably closer to like 4:15. We'll figure it out. And feel free to drop in the chat if y'all need a break, but we'll go from there. All right. So what are fungi? That's kind of the big kind of connection overall, like understanding what these organisms are, because they're everywhere. They're kind of in the background in a lot of kind of English speaking languages. We kind of tend to have this very kind of mysterious and sometimes antagonistic view of them, like, oh, mold, it's decay, decomposition, that it's these kind of undesirable aspect about our natural biology and world. But the reality in general, fungi are such a huge part of our natural world and ecology. We often think about them as decomposers, but also they're these very, very deep mutualists that basically connect everyone, and everything in our world in general. And like, they're the reason why life exists principally on Earth in the first place. So we have over here from left to right, we see a bunch of mycelium from the forest floor of a mushroom that's actually deliquesced back into the soil. Deliquescence is the process when a mushroom either decomposes after it's released its spores. And fungi are unique because the structure of the material is made out of mycelium, and mycelium is this interesting structure of individual threads called hyphae. And fungi are kind of amazing because it's a combination of like this polymorphic kind of stem cell-like system that can adapt and change its morphology and structure to suit its immediate need of its environment. And most, mycelium in general is kind of this white color, this wispy thread-like substance. And, for the common eye on the forest floor, they're pretty similar. Now, if you're an experienced cultivator, we'll be able to differentiate between different types of mycelium, they're growing out in petri dish. But if you're in the forest floor, oftentimes this isn't growing in isolation. They're actually intertwined kind of in the center. As we can see, is a mycorrhizal network. So here we have a picture of a conifer sapling within Urcoid mycorrhizal fungi. So it's pretty much a mycorrhizal fungus that grows on the outside of the roots. They're super common in conifers. And what's happening in general is that it's this deep mutualism where you have the capacity where the plant is really good at making its own foods and sugars from photosynthesis. However, in general, one can't live off a diet of like potato chips and like soda alone, like sugars. You need some essential minerals and nutrients. So that's the thing, like, everything the plant gets, aside from carbon dioxide and sunlight, it gets it from beneath the soil. And plants can pull up nutrients in general. Plants can see the nutrients in general, but the reality is you have these deeper connections where plants will actually farm bacteria and fungi. They'll have this deeper symbiosis where basically the plant will put out these exudates, millions and billions of different combinations of carbohydrates that attract certain types of microbes, bacteria and fungi, and even archaea in some capacities, to basically have this deeper mutualism. Well, they'll actually trade off So it's amazing because the bacteria, the fungi, and the archaea have these really powerful acids and enzymes. They can basically solubilize these micronutrients, macronutrients, and trace elements out of the sand, silt, and clay in our soils. Organic matter is the key part about the whole situation as well. So it's this deeper situation where you have these networks, as we can see here, that will expand the surface area of the root systems of these plants 10 to 1,000-fold. And then even further, we'll actually network and connect these organisms. Mycelium is the actual persistent organism of the fungus. We talk about mushrooms, but really that's just kind of the end of its, not really the end of its life cycle, but that's the structure that basically is a huge part of its reproductive cycle. And through capacity of actually basically sexual reproduction. So the aspect in general, this massive network, they occupy a huge surface area. We think about in general about these individual hyphae and an aspect of scale. We know that roughly they're basically a root hair, basically a root of a plant in general, around a section of it will have, I think, anywhere, roughly around 300 to 400 miles of mycelium. We talk about total kind of, length and huge amount of surface area. So it's kind of amazing how big the situation is overall, and it meshes together. So here we see some mycelium of an oyster mushroom, Pleurotus austratus, cardboard. I have a love affair with cardboard. We'll talk more about this later. But oyster mushrooms are probably the first mushrooms that I grew, and they're tenacious, and they're a great way to get people engaged with fungi as well. So part of the kind of going off this aspect, here's a nice little diagram that puts us all on the scale, is that we actually have an example of the mushroom itself as the fruiting body. That's the reproductive structure. In essence, the fungus really doesn't want to make a mushroom unless it absolutely has to. It's pretty energy intensive for it to go about basically changing its gene expression to make these specialized structures, these rhizomorphs that mesh together to make the stem and turn into the cap and unfurl. It would much rather, exist as just as a mycelial network in whatever substrate it's growing on, or mutualism with its plant in general. So the actual mycelium is the actual organism. So much like the fruit of a tree, of a fruit-bearing tree, that in itself contains the essential, seeds that will continue the progeny, basically the next generation. It's essential for that. We understand that, in the springtime, buds, flowers form. If they're a gymnosperm, they're just going to dump all that pollen out, male pollen cone, female pollen cone. If they're an angiosperm, they require some kind of pollinator, insects, birds, sometimes us in some scenarios. That female flower is pollinated, it swells, it turns into a fruit or some kind of seed-bearing structure, and then releases. The interesting about fungi is that they don't reproduce with seeds, but more so spores. And talking about mycelium and why it's so important is relevant to this capacity. So yeah, mycelium is the main structure. Mycelium is composed of hyphae, and the fruiting body is just the structure that releases spores. And we identify, fungi, namely mushrooms, because these macrofungi present themselves in these huge variety of different shapes, types, and colors, which kind of the allure of kind of getting into naturalism, this expression as well. So a kind of relation is that, think about, look at the piece of textile you're wearing on you right now, whether it's your pants or your shirt or socks. Think of that textile as like a mycelium that's composed of hundreds of thousands, if not millions or millions of different threads. Those are the hyphae. So now we'll kind of play some videos. This loads properly. Anyway, but yeah, the way fungi grow is pretty amazing because in general, they branch out in this kind of branching anastomosis. Anastomosis means something that basically connects and reconnects. We see a lot of similar kind of structure between, water crystallizing into ice, the liminal flows, in our estuaries and kind of natural water systems as well. And even to this branching nature that we see in much of our own biology, our respiratory system, our circulatory system, our nervous system. And it's kind of amazing because such a simple organism is incapable, such simple organisms, such simple structure are capable of such incredible things in general. So we really to help us understand these details. So here we basically see individual threads of hyphae growing. We have this liquid of the cytoplasm in general. And fungi are one cell wall thick, so it's pretty amazing in general. And this goes for both the microfungi and the macrofungi. And it's the variances in how they behave in all different types. So the primary structure is this mycelium composed of hyphae, and they'll grow out in this very dynamic nature, reforming and breaking down and reforming the tip of their hyphae while continuing to branch it over, which is why they continually grow. And we understand that fungi are actually a huge part of the carbon that's actually stored in the soil. An acre of healthy soil is thought to have roughly around, roughly 2,000 pounds of mycelium, because in general, it's the factor that these fungi are basically digesting the organic matter, which is basically being produced predominantly from plants. The plants are pulling out of the atmosphere and then using it to build their cell walls. And even when a distal tip of a mycelium breaks down, the shell of the hyphae is still there in place. This organism is interesting because it'll move through this kind of tube of its hyphae, basically cytoplasm, and often kind of leaving on the older parts these vacuoles, which are for sources. So it's kind of amazing how they function, really underdefining them as well. So talking about we talk about genes and expression, how fungi are so simple but incredibly dynamic. So what we're going to look here is we're going to look at a little video where they took a culture of neurospore crass. And neurospore crass is a red bread mold. It's a very common, fungus. It's a red bread mold because it's red when it shows up on your bread as well, and whatever substrate it's digesting. But it's exceptionally large enough so that on a microscope, a light microscope, we can actually see it and actually observe kind of its cytoplasmic streaming. Cytoplasmic streaming refers to the movement of the cytoplasm through the hyphal tubes overall as well. Through the hyphal tubes overall as well. Are you guys able to submit questions still? Okay, let me know if otherwise. Anyway, we'll get back to it. Anyway, so basically in general, we have a culture of neurospore crass. It's been stained, so its hyphae will be red. And then also too, it's been stained with another dye. It's a nucleic acid dye. So nucleic acids are what our DNA is made of. Our DNA coils into the compact genetic information chromatin, and which basically form our chromosomes and are stored in our nucleus. And so what's interesting is we take a nucleic acid dye and we stain it, and we can observe, through a fluorescence of these of where these nuclei are inside of the fungal cell. So what we see here is something rather interesting. We see these little dots kind of moving through the actual hyphal network. So it's pretty interesting in general, it's what's going on. Like if these are nuclei, right? Like we usually when we think about cell biology, we think about a plant or animal cell, or even a single cell organism. Okay, like one nucleus, right? It stays static. Not so much. Fungi are multinucleate. Now, some of those nuclei are staying inside of the actual septated section of the hyphae, kind of like bump back and forth, like someone trying to do like a 20-point turn in Manhattan during rush hour. But some of them are actually kind of these shuttles and schools of basically these schools of individual nuclei. The dots are nuclei. So the dots are the actual the organelles that store the genetic information inside of it of the fungus. So what's interesting is that these nuclei are streaming through the actual mycelial network of these hyphae, and it's amazing because in general we think about the aspect in general is that, okay, usually one nuclei per cell, they're static, but here we have cytoplasmic streaming where they're multinucleate organisms moving them through them. So what's going on here? When we think about like a fungus growing through its environment in the woods, it's constantly trying to survive against pathogens that are bacterial or viral, even competitors, different competitor fungi, and even mushroom-eating fungi-eating fungi. And then even trying to break down new food sources, new sources of substrates. So it's interesting. The colors, the red are the hyphal tubes, which are the substructure of the mycelium, and the green dots are the nuclei. Those are the organelles in an organism that store its DNA, its genetic information, which are essential to produce the proteins that go on to basically become the building blocks of the biochemistry that makes all this happen. So it's amazing because this organism, normally its environment, is constantly being stimulated with environmental pressure and stress. So what's amazing is that inside these little distal tips, they're experiencing their environment through chemical kind of signaling. Like chemistry is the language of biology. We don't think about it because so much of our linguistics is based around sound and also from visual capacity. But in reality, a good example, think about our olfactory system. Like scent is one of the most powerful senses related to memory because it's a direct correlation. This is kind of a similar capacity how these fungi are almost smelling with their distal tips and kind of smelling the soup of biochemistry that's going on in kind of the soil, and then basically figuring out what genes they need to express to survive. And when they figure out a way to either basically produce an antibiotic compound, something that basically helps them fight off the virus, something that actually helps them fight off the competitor or fungal pathogen, but also a range of acids and enzymes that help maybe digest their actual food source, and even communicating kind of the signaling with their plant mutualists in general. They'll relay this information throughout the mycelial network, which makes them incredibly dynamic. The DNA are inside the green dots, which are the nuclei, and basically you have a streaming where they're basically sharing this information. So much like I send someone an email like, oh, hey, I figured out a new neat trick to solve this problem. I can share that over email or social media, and it's far easier to transmit as well. Because if you think about this organism it's a decentralized network, and much like how our internet or our biological systems in our body have this some kind of fractal of kind of branching anastomosis nature. Cool. So here's a nice little slide which breaks down the kind of details of the the general reproductive cycle as well. So the reality of fungi is they're pretty interesting because in general, like we talked about, let's say down here in the mycelium, the fungus really doesn't want to have to make a mushroom unless it has to. It's pretty labor intensive, energy standpoint. Think about this. Imagine if you were imagine, okay, cool, I'm going to head upstate. I've got this really cool cabin in the woods, and I've got it fully stocked for a month. In general, you'd want to just kind of hang out and just chill in the cabin and have a good time. You wouldn't want to go back to work. You wouldn't want to have to go somewhere. You just want to stay in your local environment. So much like the aspect too is that the fungus really only produces a fruiting body when it's fully grown through most of its biomass, a critical mass of it, or it's hit a boundary. So the reality in general, it's kind of, we don't really understand the trigger that caused this fruiting procedure. Fruiting is when a mushroom is basically being produced. But it's the equivalent of like basically the fungus has sensed its local environment that it's pretty much, grown through its food source. It's hit a boundary, or usually for a number of the seasonal mushrooms that we observe, there's some kind of trigger microbially, some kind of chemical trigger that happens in the soil. We're learning a lot of certain types of mushrooms. This may be the reason why they're certain seasonal and why they exist in certain conditions. But the reality, the mycelium would want to stay as mycelium organism has to. Okay, cool. So you're upstate in your cabin, having a great old time. All of a sudden, this like crazy blizzard comes out of nowhere, like once in a century blizzard. You're stuck up in your cabin, and you're like, okay, crud. Okay, I'm here, but I'll have a month's supply of food. After like 3.5 weeks, you kind of got to make a decision. Like, you're not going to stay in your cabin, you would probably need to venture out into the snow and survive. So the reality in general is the fungus is trying to expand its network. It's trying to basically continue and keeping going. So the evolutionary process of producing a fruiting body which releases spores is this aspect. So we see the fly agar, the Amanita muscaria, the classic red white mushroom. It makes this little bud called a primordia. This is an indeterminate fungus. This is a determinate fungus, meaning that as a defined form, if you nick it, it'll be affected structure. But it grows up and basically continues, and then basically have these on the gills. If we zoom in, we have these. We have the cross section. Now, this is a Basidiomycota. This is a cap and stem mushroom. We'll get into the details later. But all along the gills are these genetically rich areas called basidia. So on the cross section of this basidia, you have basically two nuclei that are basically kept in there because they're multinucleate. That's the minimum amount that are actually inside a hyphae for most capacities, simply speaking. And we have this process called karyogamy. So it's almost like a not like a fertilization, but these two nuclei fuse. and then basically go through the aspect of meiosis where they do two meiotic divisions and basically go on to make these daughter cells that become spores. So these spores in general grow on the tip of these spore-releasing structures. We'll get into the details between like Ascomycota and Basidiomycota. But in general, the aspect. So these distal tips will release into their environment. And what's really miraculous is that, the fungus itself, the actual fungus that's been able to make a mushroom, it's not karyotic. It has basically two sets of, it is those pairs of chromosomes in general. Like we know the aspect too as well that, we get one set from our parents. We get two sets from our parents, one from each of our parents as well. Now when this primary spore lands, it's monokaryotic. So meaning that it can't go on to make a fruiting body. It's unable to recombine and go through this karyogamy. However, it can grow and digest and survive, but it's monokaryotic. It's haploid. So basically, be growing through its environment and up and goes through this process called plasmogamy, when these two primary mycelium, these two primary hyphal tips from two different spores, will grow through their environment, and then they'll sense and see if they're genetically compatible, and they'll go through the process called plasmogamy. So plasmogamy, sorry, wrong one. So plasmogamy basically looks like this in general. So you have basically these two haploid mycelium in the first section right here. So we see the one with the white dots. Those are the two nuclei. The one with the black dots, nuclei. They're able to grow together and sense that they're potentially compatible, and they'll have this bridge, and they'll mesh their two hyphae, and they're basically in general exchange this genetic information. They'll then go through the process of basically becoming diploid. So it's amazing because of plasmogamy, you have this heterokaryotic mixture, and they'll basically go on and basically to make this heterozygous recombination, and then they'll continue to grow into these new branching structures. So it's amazing because they'll recombine the genetic information. It's almost like imagine if your gametes, when we talk about human beings, gender is a huge conversation about detail, but when we say just sex with reflection of gametes, like sperm cells and egg cells, imagine if the fertilization that would normally happen inside of our mother's womb, inside of fertilization would normally happen inside of our mother, is happening actually in the environment after our gametes have germinated and grown through the environment and then had to find each other. That's pretty crazy. Like that's wild. That's why I study fungi, because that stuff blows your mind, Yeah, so and so we see here from section 4 to 5, we see these growing structures. This is how the fungus actually grows. So when you saw that video of the fungus growing through its environment, we basically see that these recombined, we see this clamp connection forming. So basically the fungus grows, make this distal tip of its hyphae, folds back on itself, and basically ensures this recombination. So you have this heterokaryon situation. So cool. So let's go in the aspect 2 is, yeah, same bro. Yeah, fungi are crazy. That's why I study them. Like Anyway, so Basidiomycota. So how do we talk about the fungi that happen to grow, evolve to, that we interact with on Earth? So Basidio refers to the basidium. They're the spore basically producing structures. So in general, you have the whole process where they're essential for basically fertilization, go through meiosis, basidiospores fall to the ground, they grow primary mycelium, plasmogamy. So this is how we talk about them. So these are your cap and stem mushrooms, the one that also have your gills. So here's a little more detailed example so you can kind of see it as well. Cap, gill, basidium spores. Those little nodules, but those little bumps between the basidium, those are called cystidia. I think last time I checked, we think they store the waste products from the metabolic process, which is pretty important. You wouldn't want to have like a garbage dump next to your nursery. So it's pretty important from a biological capacity to maintain that. And then also too, like a lot of the fungi that we see in nature, a lot of these brackets, a lot of these woodier conchs, a lot of medicinal fungi like reishi or turkey tail, they have these pores. So rather than having gills, they have these pores that are studded on the inside with these basidium that basically fall down. So we can see here, we see fomentarius, we see a cross section over here. We see the pores, and then if you actually look at the bottom of the mushroom itself, that's actually where the spores get released. And then we have different iterations between different species of fungi. So these are 3 different types of mushrooms, Trametes, 3 different genuses and their respective species represented. Trametes, Trichaptum, and Fomitopsis all have different iterations and patterns, and these are really some amazing and beautiful structures and patterns. So it's amazing, really kind of just how beautiful they are from like even from an aesthetic standpoint. That's kind of, I think, quite a big appeal why they're kind of make their impact right now. So let's zoom in. Cool. Enhance. So we see a scanning electron micrograph of how big these are. So we see a field of Basidia and Cystidia kind of all over the place right now. And then we see roughly the kind of little cluster. That line is 25 microns. The actual head itself is around 10 microns, and we see how basically big that is. So Basidiomycota refer to, so Basidia are on the gills. So they're the structure, the surface of the gill, we call this the hymenium. We see the cap, we see the gills. On the gill is the hymenium, and then studded along the hymenium are all these Basidia. And then if they're actually a shell fungus, those rather than on the surface of the gill, they're going to be inside of each of these little pores. So that's kind of the aspect as well. So how do these spores get liberated? How do they sporulate? How does that actually happen? So what's really amazing is that, we think about the environments that, that fungi actually, where do we find mushrooms? Like we often find them in these kind of really moist environments. Just after it rains, just after the soil has been hydrated, the water is starting to evaporate. And we have this kind of microclimate on the forest floor. So it's really kind of interesting, actually, what happens. What's the actual mechanism that allows this to happen? So what's interesting is we see here in this example, we see a sterigmata. So a sterigmata is the actual little distal tip of the basidium, where the actual spore develops. And then we see the sterigmata, the spore. What will actually happen is that the fungus will produce a range of sugars on the distal tip of the sterigmata. And what will happen is this will basically, in the humid environment, this will attract a formation of basically a water droplet to form. Now, mind you that, we think about a microclimate going on. We know that, when it's humid, basically you have things evaporating and we have water condensing. So we basically have the water condensing on the basidium, the cystidia, the sterigmata. And what happens is eventually this drop of water grows. It's called a Buehler's drop. And what happens is we know that, due to surface tension, if you've ever taken a glass of water, filled it up, and you have that dome over the top, that dome over the top, it's called surface tension because water, the essential, molecule for life, has hydrogen bonds. So water molecule is like polar. It looks like kind of like someone doing a shaka like this with your pinky and your thumb out. So your pinky and your thumb, those are the hydrogen. Basically, those are the hydrogen atoms. And then the oxygen is basically your knuckles and your fist, and they basically bond to one another. And what happens is as that water droplet gets bigger, the attractive forces between the water molecules will grow. And what will happen is the water molecule, that Buehler drop, will become attracted to the water molecules on the actual cystidia or basidia, and will pull down. The kinetic energy that happens when the water droplet forms is enough to launch the spore off. So we see that launching motion. What's amazing is that often the forces that these occur at are roughly 20,000 Gs. Like we're talking about spores landing centimeters to inches away from the actual mushroom itself. To give some perspective, astronauts, when they exit our atmosphere, they have to achieve they're often experiencing 20 Gs. So that's 10,000 times as much. So it's amazing that these organisms have adapted such a unique structure to release their spores. And then even too, we understand that some of our truffles or some of the tubers are actually mushrooms that have actually evolved to the point where they've lost the ability to release their spores. So rather than relying upon a microclimate, they will just basically produce aromatic compounds that will attract certain animals to basically come and eat them. So it's amazing how this whole process is. Cool. All right, so that was a lot of information. I guess you could say like, well, no, like, so tuber. Okay, to clarify, um, off we refer to potatoes as tuber, and then also truffles. Some of the genuses refer to tuber, but because they're under the ground, and like in French, the word for like potato is like pomme de la terre, which means apple of the earth. So language is confusing. Uh, anyway, yeah. So we have Ascomycota. So Ascomycota, you know, they're everywhere. They're probably the ones that we're the most familiar with, whether we like it or not. They are your molds. They're basically they do make these macrostructures, but the majority of them are these micros. So if you ever got a let's say your partner, your relative, your roommate that has a little science experiment going on in the kitchen, maybe in the refrigerator, you know these are the ones that show up to the party. These are the microfungi predominantly. Some of them are macrofungi as well. They're often referred to as the cup fungi, but the majority of them are actually are these microfungi in general. So we call them Ascomycota because the structure they release their spores are these asci. So if we look over here, we see this cup fungus as well, and then we see these little kind of tendrils. So these tendrils are all studded along the inside of the actual cup. So whereas we required a microclimate for basically a cap and stem mushroom to release its spores, these just require a bit of disturbance of kinetic energy. So it's very slow. That's why when you see mold growing on your bread inside of the bag, it's too late. It's already all over the actual system as well. And if anyone's ever seen a puff ball and you've tapped it, basically it's relying upon the kinetic energy transferred from an animal or any kind of falling water or any kind of falling debris to basically hit it and release these spores. And they basically are packed into these little asci, these little fingers. It's almost like a cork bat. So if anyone who's into baseball, or even if you're not into baseball, so if someone's playing baseball, you often want a lighter bat to get a faster swing. Oftentimes, if someone gets caught, the bat will split open and the cork will fly out. So almost like corks inside of a bat, they'll fly open and be dispersed. And so they'll be packed with spores, sometimes 4, sometimes 8. Ascomycota are crazy. But yes, they also go through the process of basically going through the process of kerogamy, meiosis, mitosis, primary germination, and plasmogamy in the whole process as well. So here's an example of like a cup fungi that we have. So we see here that the mycelium in the bottom, we see it mesh into a rhizomorphic structure, into its actual fruiting body. And then we have the whole structure where we actually have the sterile hyphae in between developing and mature asci to support them. So it's this amazing structure of how they develop into this form. So we see here, so we see some asci from a slice and section. And then if anyone likes morel mushrooms, this is a image of Morchella spores, and they're actually incredibly beautiful. And so microscopy with fungi is a whole another world where you can really dive into these intersecting connections. I'm gonna just turn off the question thing, just because y'all put them in the chat, which is great, and I can see it. So yeah, we'll just go from there. Cool. So yeah, alrighty. So a bit of like fungal biology, kind of breaking it all down from where we started. We have on the upper left here, we have an image of a generalized range of hyphae forming into the distal tips. And so we have a bit of cell biology. We see that they're one cell wall thick. Inside of them, they have a cytoplasm where all their organelles are stored. Their vacuoles, their nuclei, their ribosomes, their yada, 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 full on cell biology, but they're multinucleate. So that's the key thing to keep in mind, because they are going this they're going for this heterozygous situation. The distal tip of the hyphae is called a Spitzenkorper, which is an amazing word, but it really refers to the fact is that this hyphal tip, when it grows, it's almost equivalent to like you have like a party balloon and you're filling it with water, and that basically it's continue to grow, but when it reaches that maximum point, it's going to basically doing that clamp connection. What's happening with the hyphal tip is that the wall of the hyphae, the distal tip, is being broken down and reconstructed simultaneously. So it's kind of mind-boggling, this whole structure that functions over and over as well. And they're one cell wall thick. So we see the top right, we see a generalized explanation of what that hyphal cell wall is. So the outer layer are these manoproteins. So it's a mannitose, a range of sugars bound to proteins, so they call them glycoproteins. And then basically that's kind of like the pellicle, the outer layer right there as well. Then we have these alpha and beta glucans. So these are these large triple helical sugar polymers of sugar that basically branch and twist together to basically act as like the structural girders that actually hold up the cell wall itself. These are some of the actual these are some immunomodulating compounds. We'll get into them as well, but alpha and beta glucans make up this well. Then we have chitin. Chitin is the reason why for the majority of the population you can't eat mushrooms raw, because it's a really dense polymer. It's a acetylglucosamine. It's pretty much, it's pretty close to cellulose, but it has a couple extra nitrogens and oxygens thrown there in general. So that's why you always have to cook your mushrooms. I think a very small amount of the population has genes for lignases. For chitinases, and even small animals do as well. And then we actually see the inner phospholipid bilayer. So these little balls, it's a phosphate. And then we have a hydrophilic phosphate on the outer section. We have a hydrophobic, basically tail. And then we have a hydrophilic phosphate. So the phospholipid bilayer is like the foundational basis for a lot of animal cells. So what's interesting, we see an example of like combination of like somewhere between plant and animal, and it's really interesting because fungi are actually more closely related to fungi than we are to plants. And we'll kind of get into a bit later as well. And then we have membrane proteins, which are basically in place, which is a huge part of these proteins that basically sense and receive and facilitate a number of active transport procedures. Cool. All right. So on the bottom, we see our Ascomycota represented by this Penicillium species. And then on the right, we have our Basidiomycota represented by this oyster mushroom, Pleurotus austriatus as well. So what's interesting is that we often think of like the most efficient spore producers being like these macrofungi, because these big mushrooms are loaded with Basidia, they're Basidia, and they'll release them all the time, but they require this microclimate to fruit. In reality, it's the Ascomycota that actually are really like top notch at making this basically happen. So that's why Ascomycota are prolific. So what do fungi do? So we understand them primarily as, the decomposers of the world. They're able to basically break down and digest organic matter, and even in some cases that we actually understand now, actually make bioavailable inorganic matter, take inorganic matter and basically solubilize it into a number of bioavailable forms, making micro, macro nutrients and trace elements available for life to basically happen. So we see a progression of a substrate, quote unquote, some wood chips that have been hydrated and then been inoculated with mycelium, and oyster mushroom mycelium. And so this basically grows, and because as the mycelium, the hyphae grow and the mycelium reaches critical mass, we can actually visually see it, manifest in this white structure. That's why you'll see it usually if you're out in the forest hiking along, and you see when you have mushroom at the bottom, these little kind of threads. Even to let's say you're mulching, you'll see them growing. And if you're an even indoor cultivator or anyone that messes around on cardboard, you'll see it. But eventually the factor is this goes on to basically become food for a variety of microorganisms. In general, nematodes, microarthropods, because the reality is these fungi are able to basically do a lot of the heavy lifting, because we understand fungi to be primary, secondary, and tertiary decomposers, and oftentimes they're working in unison with all these different microorganisms. So this is kind of the soil food web. So this was described by Elaine Ingham in her monograph paper back in the 1980s. And the reality in general, it talks about what's going on in the soil is that it's not just a solid state thing. It's very much a biological process. It's a whole ecosystem going on underground. Basically, in general, we understand the first trophic level that they're plants. Plants are able to basically make their own food. They're able to make their own sugars. However, you can't just live off, like I said before, potato chips and soda. You need some trace elements and minerals. So as we're understanding that as plants moved onto land, it was the combination of these first, these primitive lichens, these symbiosis between basically fungi and photosynthesizing bacteria or algae. And then the reality over time is you have the symbiosis get turned inside out. So fungi are really important because they occupy the second trophic layer in the food soil web. They basically are able to, with bacteria, solubilize, and archaea too, solubilize inorganic micro, macro nutrients and trace elements and trade them for plants. And then the resultant stores left over in their bodies are basically consumed by a range of nematodes, arthropods, protozoa, which basically poop out plant available nutrients, which are the foundational basis for that. So fungi are able to decompose effectively in this factor. So here's an example of New York City soil. It's really sandy. Yeah, we have really sandy soils because glaciers that were a mile tall ground down these ground down the limestone and other kind of rocks into basically the sandy soils we have today. However, this is an urban soil, and we see here these little granules. And if I was to play a video that I recorded this, which I don't have on here, you would see it pulsating because we see a bit of organic matter here. These fulvic and humic substances, which are these macromolecules of organic soil matter, but also too, it's full of bacteria. So a lot of our urban soils are bacterially dominant, and we know this because a lot of plants thrive in these environments. So we understand that fungi tend to prefer more acidic soils, and fungi are a huge aspect for increasing the organic soil matter content as well. And this is a whole range for profiles of different plants. So this was some work I did with MS-126. I was invited to basically be part of their soil cycle program. It was a program that basically teaches youths about soil and how it functions, the biological aspect of soil. I had the opportunity to talk about soil biology. That was Faye Walker who invited me. She's an amazing woman. And really, we have this aspect of teaching middle schoolers over in Greenpoint about urban soils and soil biology. And this was funded as part of the Newtown Creek Alliance. Well, part of the settlement from the Superfund site that was settled with Exxon between New York State and the EPA. So it's highlighting the work we did in general. I'm just talking about the Greenpoint Community Environmental Fund. So some work we did soil building. So we kind of did some lasagna gardening. It was a little chunkier back then. But in reality, in general, we talked to kids about soil biology. So we integrated like presentations in the classroom. We integrated hands-on exercises with raised beds about how do you actually build and compose soil overall. So these are some raised beds. The idea was we buried them. Basically, I think it was back in we did microscopy on them back in April. We inoculated we did microscopy like in March. We inoculated them in April, and then basically the mycelium grew. We did a lasagna mulch bed of mycelium, cardboard, sawdust, and just layered them, and then just basically just dressed them with mulch on top, and then basically had the opportunity to for it to grow and connect, and then these huge mushrooms basically fruited out at the top of the raised beds, and we didn't pick them, we just let them sit. And the aspect too is that really was to basically demonstrate an example of ecological succession that actually happens in nature. You know, you imagine a tree, it grows through its whole lifespan, it gets old, it gets whittled out between insects and mammals and bacteria and fungi. Your primary decomposers breaking it down, it falls, secondary decomposers that are more complex living species, and tertiary decomposers. So we kind of simulated a successional biology that goes on. So these mushrooms fruited, but rather than harvesting them, we had let them kind of come to term, and they started to decompose, and that attracted insects. The insects laid eggs, the eggs hatched into larvae. Larvae attracted different types of birds. They basically drop seed, and you have this natural restoration of ecology. So it's basically, how do you understand that with building soil, you're trying to mimic a process that actually happens in the natural environment, because in an old growth forest, trees grow and fall down and decompose, and you have this whole balance of the carbon and nitrogen cycle happening as the way it's been played out over several billion years. So little intro to soil ecology aspect. You know, dirt, sand, silt, and clay. Soil is not dirt. You know, you need biology and organic matter to turn dirt into soil. And depending upon where you live, you'll have different ratios of sand, silt, and clay. And that actually you can simulate a loam, which is a quote unquote concept of a equal distribution of sand, silt, and clay by, you know, using biology overall. So food soil web, this was for the kids, you know, a nice little illustration, you know, cartoon situation, which is good. Plants, they do a lot of the work, and it'll die back as biomass. Bacteria, fungi, protozoa, nematodes, arthropods. And yeah, this was an example of, after the this is an example of after the actual process, we see a soil sample now. Basically what this is we see there's way more structure in the soil. It's not just densely packed together like sand. We see there's organic matter floating around it. We see there's bits of these humic and fulvic substance, plant debris, organic debris as well. But also we see that, these fungi are having a huge aspect of binding all these microaggregates together. Now, basically, bacteria will make these alkaline glues and bind to sand, silt, clay, or organic matter. And then basically you'll be having the fungi, which are these girders on a bridge that actually pull it all together to make this macro aggregate. So this is basically how soil structure is built. And we can actually see these individual hyphae coming out of a root tip, and what the septa is closed as well. Or aspect, the install we did in Manhattan Avenue Park. It's this National Wildlife Federation. So it's Greenpoint, it's Eco School Sustainability Toolkit if you want to look it up. And kind of it's where I think for this part of the presentation we can kind of transition into the aspect of what is our relationship with nature. And I think it's a serendipitous time right now with everything going on, that it's not business as usual. That reality is that we're very much part of our ecosystem. We're not the top of it, and that there is a large ripple effect based upon what actually happens and what influences we play. That it's not ego, it's eco, that we're part of a large dynamic system overall. And this kind of goes into an aspect of applied mycology. Cool, all this information about identifying mushrooms and learning maybe the molecular biology and cell biology, that's cool, but that's great. So how do we use them? How do we understand to work with them? How do we implement concepts of biomimicry to really acquire the benefits and bounties that these fungi have overall? So this is kind of a nice little wheel. The image is from Paul Stamen's Mycelium Running. And he has a diagram further on when it comes to micromediation of this kind of flow chart. I modified a bit. And it talks about, fungi, working with fungi has this very circular dynamic process to it, organic matter is necessary for growing mushrooms. This becomes food and medicine, not just for us, but all different types of organisms, and basically the chemistry of basically the different chemical compounds that are present in their edible fungi that are beneficial, advantageous. This biomass left over from fungi is basically great for composting, is amazing for soil health, which builds living soils, which adds organic matter and microbial diversity back to soils. This makes fertile soil, which can very easily grow plants and trees where they're not stressed, they're not producing this chemistry which tracks a microbiome which is pathogenic. It's more of a mutualist, and then this in turn produces more organic matter. So it's a natural process of this circular dynamic. And we're realizing now that that human beings we're the only animal that really wastes. Waste is a design flaw that was kind of invented by humans. So it's important for us to understand that relationship overall. So I kind of got into messing around with mushrooms and growing mushrooms from, just growing out, oyster mushroom mycelium on coffee grounds and cardboard. That was the first experiment with mushrooms I had, even before Petri dishes. And it was interesting because I was working at a cafe and a bar, and we had plenty of cardboard from any boxes that came in. And then also plenty of coffee, and I had this mycelium, and it was super easy to work with in general. So it was really amazing because I could witness this biological process of metabolism happening in my kitchen, and I was understanding concepts of pasteurization, understanding that I could try to mimic the conditions that actually happen. I didn't really need a whole flow hood or sterile details. It was really easy for me to work within the confines of understanding what conditions I can facilitate the fungus without trying to isolate it or try to get it into a super sterile environment. Oyster mushroom mycelium is great. It eats just about damn near anything. And we see these pools of liquid. These are metabolites. They're kind of basically digestive acids and enzymes. So we talk about how, the fungus is basically producing these metabolites to break down, the organic matter, to make this macro aggregate. So this is basically how the fungus is basically producing these metabolites. So we talk about how the fungus is basically producing these metabolites to break down, the organic matter, to make this macro aggregate. So this is basically how the fungus is basically producing these metabolites to break down, Basically, it's biomass, but also produce antiviral compounds, antibacterial compounds, antifungal compounds, basically to protect itself. So it's swimming in its own digestive enzymes and antibiotics. So the reality in general is fungi, if you think about us, like, we're more closer to fungi than plants. We developed a digestive tract. Our common answer to fungi is whereas we branched off, where we had a primitive stomach, where we enveloped food or organisms we ate, and we digested acids and enzymes. And as we evolved, we got more complex in, how do you distribute nutrients around this digestive tract? Whereas fungi are kind of so simple because you see this branching and massive structure that exists throughout our biology, but is turned inside out because they're so simple because they basically digest externally. We ingest, then digest. Fungi digest and ingest. So you can see the confluence as it's growing over and over. And this was over the course of like 2 months. I didn't really know what I was doing back then. I was experimenting, and I didn't realize how important keeping it dark and keeping it warm was important for incubation. And, and that's the reality. I started noticing these little mushrooms, these primordians. So these are the little kind of baby mushrooms that form. So the first couple of slides were over basically a month or two. And this progression, just over a week. Amazing that, you can produce something off what was ostensibly, arguably just waste, that it was going to be just thrown out and basically ended up landfilled or in producing methane or incinerated and producing carbon dioxide. When in reality, you can make dinner. And so it's important. The reality is we can not only make bioavailable nutrients, we can take fungal bodies and make, through their substrate or their fruiting bodies, make food not only for us, but also for plants and the microbiology in the soil. And the reality in general is that fungi have this capacity of being having such a huge range of metabolic enzymes and acids and produce all these compounds through their unique genetic arrangement of how they express information and how they survive in the environment, that it allows them to kind of have almost kind of this mystical or magical kind of effect, but it's this really elegant biochemistry in play. And then when it comes to working with this, if you're going to grow mushrooms or on coffee grounds and cardboards, you'd have to think, okay, where are these coming from? What source they are. But when it's coming to making plant food or compost, you can be a bit more liberal. If you're going to make human food or animal food, I'd be a bit more conservative about understanding where these sources come from. So yeah, the kind of the wheel of growing mushrooms, food, and medicine. So kind of a nice little snapshot back over there for a while. So also too, so I had some friends that were artists and architects. They approached me because they were interested in doing an art exhibit. They wanted to make these totems of mycelium and basically hang them from the gallery, but they only had, roughly 2 to 3 weeks. So with bioart and working with biology, you're often working on its time. We tend to have this bias of getting things done now because most of the things that we interact with in an artificial sense they're inorganic or they're static or stable, and we can very easily manipulate them. But biology is, it does its own thing. You can work with it to some constraints, and you can even say agriculture is humankind's ability to kind of coerce or coax nature to get it to do what it wants a fair amount of the time. So it was interesting in general. Artists were Billy Pate, Lily Jackson, and Franklin Romero. Those are 3 of my friends. So my friend Franklin is an architect. My friend Lily, she's an artist and also an incredible community organizer and great passion for the environment. I think Billy Pate is a formal practicing artist as well. But yeah, so they wanted to make these columns, so we didn't only have time, so we packed hundreds of pounds of spent, quote unquote, spent spawn from mushroom cultivation, cardboard, coffee grounds, sawdust into these aluminum air conduit tubes and let them incubate. And basically, in general, you have this whole capacity of you have this whole incubation tube you could pretty much, they wanted to hang. They only had 2 weeks, so they didn't actually work in that capacity as well. So the reality in general. Was that, they kind of, when they presented them, okay, we'll just lay them up. They kind of look like a nice little geotextile thing. We kind of crimpled the spawns. It was really funny. This art exhibit was like, It had a really interesting aesthetic. Like it was like, oh, earthy, and this like kind of burner kind of vibe in general. And we had this installation, and then the people were like, oh, it's gross, it smells. And we're like, it just smells like compost. Like it smells better than compost. So it's really kind of funny, like this aesthetic, it got in the way of the aesthetic, even though it's probably the closest thing to nature in the actual gallery space. So over the course of the exhibit, Franklin worked nearby. It was in Chelsea. He came down and he would miss the mushrooms, miss the mycelium, and then over time, we started to see these mushrooms emerge out of these totems. So it was really amazing because a lot of people were really blown away. You know, they were like, oh, what was so disgusting and putrid and vile? This whole preconception of fungi or death and decay and rotting became something that was beautiful. And that's what's kind of amazing with fungi, because they really are a non sequitur. They really catch people off guard with how their whole what their biological capacity can do, and they make these amazing fruiting bodies. Elegant, like just truly elegant. So yeah, definitely. So this kind of brings into the talk about, with applied mycology, microrestoration, right? We see fungi, we can work with fungi, we can grow food with them, we can even help facilitate the breakdown of biomasses. So we kind of focus on this aspect, from food and medicine to compost biomass, to building living soil that can grow plants and trees that can basically produce organic matter. So it's elegant kind of interconnected nature. So this is a great little snapshot talking about the Amazon effect, that we're drowning in cardboard. I have a love affair with cardboard. Talk for hours about it, how it's the best thing in the world, but we just throw it out every single day. And the reality in general is that, you know, the way we used to deal with cardboard was that we would just ship it across the sea and someone else would process it and recycle it. That kind of stopped recently. A bunch of nations said, we're not taking your cardboard. There was a practice where American companies that facilitated this would just pack raw, like solid waste inside of a pallet of cardboard, which was just horrifying and terrible. So they said enough with it. So we're swimming in it at the moment. So cardboard's everywhere. So it's a huge problem. How do we actually break it down? This is my friend Blackie. Blackie's awesome. I met Blackie as part of the community biology in New York. Blackie's amazing. Had amazing work with mushrooms when he was in college. Really passionate, really close friend. Hey, Blackie. So Blackie's holding a life box. Life box is, I think, one of Paul Stament's projects, where he basically, in the water where they would make cardboard, they would release spores into it. So when the cardboard was formed and dry, the spores would be dormant, ready to germinate when decompose as well. That's really cool, but it's the reality of trying to get infrastructure of carbon production at scale. To do that maybe is not as practical. So the reality in general, when we think about what, you know, this biomass is made of, you know, it's made of cellulose and lignin. You know, the reality in general is that nature has gone through the process of producing these compound chemical structures. You know, this is terrifying. I'm sure organic chemistry, run, hide away. I'll make it simple. But the reality in general, the cellulose on the left, pretty much identical to the kind of thing we're wearing right now. We're all wearing cellulose or some cellulose blended product in general, because, you know, cotton is a plant that basically makes a nice little puff of it that we pull the seeds out of. It's a whole different story about agronomics and detail and history, but keep it simple, cellulose. Then lignin. Lignin is what makes wood. It's basically this heterogeneous, basically polymer of all these different structures that basically binds together. We have a hypothesis that during the Carboniferous period, you know, basically that, you know, plants figured out a way to evolve lignin, and basically, you know, they could grow higher than a meter. They could grow higher than 3 feet. But also, too, at the same time, bacteria and fungi at that time in human history, in natural history, bacteria and fungi at that moment in natural history did not have the acids and enzymes to break lignin down. So you had a period for 300 million years, we think, that trees just grew and fell down and nothing could decompose them. And actually, it's where we think a majority of our coal deposits come from. Most of our petroleum deposits come from algae. Most of our coal deposits come from these trees in this Carboniferous period. Carboniferous, coming from the production of carbon. So, you know, fungi are really good at breaking these things down. Here's the connection. So, you know, you had a nice little plant. You got a plant cell wall. We zoom in on the cell wall. We see this kind of interlacing of cellulose wrapped in hemicellulose, and that bonds to lignin. This looks awfully like a building structure. It looks like kind of like girders and rebar with all this kind of webbing in between. And it's amazing that, nature evolved these structures to hold things down. So it's resilient and flexible, and, that corn plant or where a plant can grow but also sway. We don't think about trees as motile. Well, not really motile, but that they're static. But if you ever seen on a windy day a tree its trunk will stay in position, but its branches will gradually sway in accordance in that capacity. This is given to the structural nature of how plant cells with the structural chemistry of cellulose, hemicellulose, lignin basically are able to elongate and extend. So the reality in general, cardboard's everywhere, and these fungi, they're primary decomposers. Well, primary decomposers are the first stage of the fungi that do the heavy lifting. They do the wrecking crew that comes in there with a wrecking ball and has these really powerful acids and enzymes that can break down the complex structures of cellulose, of lignin and lignin, cellulose, hemicellulose. And then different types of fungi will even selectively decompose different types. You've ever been in the woods and seen a log that was all white? It was wispy. That was a white rot fungus. That was a fungus that predominantly decomposed the lignin. And then if you ever seen a tree on the ground that's blocky and cuboid, this rot, that's a brown rot fungus. That's a fungus that's selectively evolved to basically decompose cellulose, to basically be easier to break down as well. But we see this mycelium in general loves cardboard because we've done all the hard work for the fungus. We've taken that tree, we chopped it up, we've macerated it into a pulp and extruded it into different shapes and forms, and it's everywhere. So this is an example of when I teach workshops, I often do a cardboard lab experiment. So basically taking a bunch of spawn. Spawn is the word we talk about whenever a substrate or biomass the fungus consumes. A macrofungi, predominantly we're talking about, grows through it and then has reached confluence that you can basically take it and then inoculate it or transport it to basically grow into new biomass. So I think the exercise is where we take cardboard, inoculate it with oyster mushroom mycelium, and then basically it grew out to this log. One of my attendees put it into a Tupperware container. You can see this mycelium mat grow over the bottom of the actual Tupperware container. It's more so growing over it. It's trying to find new food source, and you can see the medicinal metabolites as the mycelium network is basically digesting the biomass that is still present actually in the mycelium log. So yeah, love affair with cardboards. The reality in general is something in cardboard. So thinking about how mushrooms are getting more popular, we got more cardboard than we know what to do with. A lot of the situation we do now is unless we recycle it, it's often going to be landfilled or incinerated. And actually we're lucky to live, at least I'm lucky to live in New York City, where we do have a paper mill in Staten Island. We actually do recycle a fair bit of the cardboard. But the one little thing is that, if that cardboard is contaminated with food, grease, or oil, the cardboard processing equipment can't process too much of it because it'll make a bacterial biofilm. It can make biofilms that attract other microbiology that will damage the machinery and cause it to repair and affect the productivity of the plant. So oftentimes cardboard that's too contaminated will just be landfilled or incinerated. So the reality in general is that fungi not only have these macrofungi, these oysters, mushrooms, not only have acids and enzymes. Enzymes that can break down the cellulose and lignin, but also they have lipases. They have the actual enzymes that can actually break down the fats. And this is a huge aspect that, fungi are able to break down fats through one of the unique kind of situations in that capacity. So it's amazing, like break down the cellulose, the lignin, the hemicellulose, and the actual fats. And if it can't break it down, it'll break it down or arrange it to a point that other microorganisms can actually digest it and consume it. So this is the thing about it. We tend to think with micromediation that's, it's just fungi. And I thought that was the way too, but this is kind of the reductionist trap like we sometimes fall into, but realizing that this is a complex dynamic system, that they're all interrelated and connected with one another, serving all these different roles. So here's a bin we talked about in general. So this was Beam Center. Basically we see these, and the reality was not really trying to grow mushrooms, but trying to grow mycelium, because we're interested in composting, and a cool little macro shot of some primordia forming with some water droplets. Yeah, the aspect is that, really make a biodigester. You could really just take this cardboard, shred it up, pasteurize it with some hot water. Pasteurization is the aspect of basically heating something to the point that most microorganisms can't survive, not all. So the reality in general is you actually have some beneficial cooperative microbes that actually work with the fungus to break it down. And we see the aspect that it actually, once inoculated, basically will grow out to prominence and will actually grow out to confluence. We see the metabolites there as well. So it's interesting thinking about this. We have this kind of successional biology. This doesn't just get limited to trees in the forest. We can actively use waste streams that are a huge problem in our world, but also a huge resource and have huge value. Kind of talking about, we can emulate what happens in nature because we've actually done a lot of the heavy lifting by doing that mechanical and heat energy process by basically turning that wood into cardboard. So this was an interesting aspect. I had an opportunity to work with a high school student this past spring and summer. She reached out to me because she wanted to study micromediation. I'm like, cool, you're 16. We can't get you into a brown field. Can't do anything with heavy metals, can't do anything like that. So the reality in general was like, okay, how do we do something practical, right? How do we focus on micromediation not as like an after effect or bioremediation after effect, which is most of the field is about, but how do we divert the inputs that are normally a problem that end up in our environment as contaminants or pollutants? How do we work with this capacity? So this was a kit from Biorad. Biorad is a biotechnology company that make lots of equipment and reagents, but they also make kits for molecular biology and biotechnology education for high schools for AP biology. And they basically had this kit where for biofuels were all the rave in the early 2000s up until the early and mid 2000s. So the reality in general was that, there was a huge amount of money being poured into basically developing the science, educate people, where they had this one experiment about how do you source the enzymes that can break down the complex polymers or the cellulose polymers into simple sugars to be fermented and turned to ethanol for biofuel. So one thing about it was prospecting enzymes from different mushrooms by literally taking a mushroom, extracting some tissue, extracting the enzymes from that tissue, and then basically doing a test on it to see how effective it is. And this was a colorimetric assay. So it was like, okay, cool, awesome. Here's a kit for something I'm not interested in, but it has mushrooms in it, but actually can correlate to what I'm trying to articulate about. Okay, like we have this aspect where, the fungus is basically growing through and myceliating the cardboard and biomass we want to basically break down and compost. But it's a qualitative assessment. I can see it, but to really do good science, you need to be able to measure things. You need to be able to quantify and kind of compare things as well. So it's a cool example of preliminary study that basically integrate a high school student who'd only taken environmental science, but not any kind of advanced biology or biochemistry, talk about enzymes, talk about environmental science and environmental chemistry and all these different details. So we have a little kit where we have the different mushrooms from shiitakes, oysters, king oysters, maitake, button mushrooms, and then extracted tissue from them, and then basically understood how effective are they at breaking down the cellulose. This is the aspect of cellulose linkage of sugars. You have an enzyme. There's a whole range of enzymes, but they focused on cellulobiose, which is the one that actually nicks the two sugars together into two molecules of glucose. So the reality is you use a reagent, p-nitrophenylglucopyranose is a really fun word to say. It's a sugar. Basically it's a glucose molecule bound to this compound called p-nitrophenol. So when the enzyme action works on it, it cuts it, and then you have basically a colorimetric assay where if you put that solution into a basics, if you take that solution with the mushroom extract into a basic condition, a quote-unquote stop solution, you can see the prevalence of yellow, which is how deep a color yellow it is, which is indicative of how much enzymatic activity actually is going on. So that was a cool colorimetric assay. And then if you have equipment that can measure the absorbance, given the fact that the solution is emitting basically a yellow light, but it's absorbing violet. So the reality is that the opposite of the color wheel, the opposite of yellow is violet. So you can have a device that actually measures the light that's being absorbed. So it's really kind of cool. So you can actually quantify it beyond color. So we did the whole experiment, divide them up, pipetted them, and actually basically developed a graph, and you can see the color. So we see that the button mushroom didn't really have too much going on. So you have the labels next to the standards. And you see the king oysters, because button mushroom probably is a secondary decomposer. It's a compost-loving species. It's interacting more with the microbiology in the soil and compost than it is trying to break down the lignin or the cellulose. King oysters, maitakes, those are ones that grow on older trees. We often find them growing at the bases of stumps. And reality is they're probably relying less upon enzymes that break down cellulose because well, they don't have it. They have enzymes that break down cellulose, but they aren't as robust as other mushrooms because they're often working in unison with um, the other microbiology. And then we have the shiitakes and the oysters, which are the common mushrooms that people cultivate on logs, which are the heavy hitters. They're the wrecking crews that actually do a lot of the details. Oh, the software that was just Google. That was just Google Docs. I just painstakingly arranged the detail. But to calculate the absorbance values, I used the spectrophotometer, which was a specialized piece of equipment. There are some ways you could hack it or do colorimeters. You know, it's a way to basically actually set a set absorbance value for the way lactic light being absorbed. So, but you can see the actual presence of how effective the extracts were, versus like measuring them as well. So it's a cool way to being do qualitative but also quantitative, like visual but also something you can measure as well. So then even two, we did the aspect where we did trials. The top row are basically so with any good experiment you need basically variables and controls. So we did basically two controls with, okay, cool, like, yeah, burying cardboard, you can compost cardboard. What effect does the mushroom have on the mycelium? So we basically tried it out with so the variables in general. We had a myceliated cardboard, obviously, that was fully myceliated. We could have done gradation to see how long was mycelium, but the general procedure was that you're adding to this bin once a week. You bury the mycelium. You basically take after about a month of adding cardboard and mixing it and letting it grow, you take a third of the bin out and bury it. And then this example, we did strips. We can measure, a fully myceliated sheet versus dry cardboard sheets that didn't have any treatment, and then a pasteurized one. So one if you got it wet with hot water. So we see those are basically the controls in general. So we see, in essence, in general, we see that the whole progression of time over the whole course of the weeks, that after basically a month, you see the bottom left, you see the and then correction, we also did indoor and outdoor plots. So the top section is indoor, the bottom section is outdoor. So using potting soil versus using the soil from an actual garden or actually the nearby woods. And then the top rows, the leftmost top row was just dry cardboard raw. The center was pasteurized, and the right was myceliated. The bottom was outdoor, dry cardboard. Center was pasteurized. The right was myceliated. So after basically we did the variables for each square was an aspect of roughly time duration. So the aspect you would take a bit of the cardboard out and uncover it and bury it and compare it. So it's kind of this sideways left to right. But in reality, the thing is you can see is that the the cardboard digestion was far more effective, far more for the wet. But for both indoor and outdoor, the myceliated cardboard was completely gone after the month. The aspect too is that they were buried and unburied as well. But it was a concept preliminary documentation about how you can actually measure the enzyme activity of the cardboard with a high school student, and definitely something that could be developed into a proper experiment with more variables and repetitions to that. But that was outlined in a research paper she did. Accelerated decomposition of cellulose base paper waste mediated by saprophytic fungi enzymatic activity. She presented it at a science fair, took home third place in the sub-region. It was going to do a regional. It was going to progress on the regional, but due to the whole kind of public health environment right now, that kind of postponed the foreseeable future. But I would love to kind of formally develop this as a great way to kind of teach environmental biochemistry and practical application of upcycling. So microfiltration, really kind of covered by Paul Stamens, talked about, more so about how do you understand that this fungus is a huge surface area and potentially could break down and digest biomass and material. Well, it could potentially be used to treat anything that's moving through water. Focusing this requires a bit more work. It's the fact that that mycelium is able to grow through a large number of biomasses and materials and proliferate them incredibly well. They bind things together. They provide structure. And that the biggest issue with runoff is that, you have no structure in the soil, that pretty much things just pass right through them. And then often you get in most cases, it's usually from fertilization or, in the case with Paul Stamens, was talking about was septic tanks by the Skookum Inlet. So here's an example of a scanning electron micrograph of a whole mycelial mat, and then on the right you see basically a single hyphae, which have its own microbiome of these rod-shaped bacteria. So the aspect was that, with aside from agriculture runoff, runoff also is present as well from from gardeners. We actually, I think, gardeners use 3 times as much fertilizers and pesticides than agricultural does. So most of the runoff actually is predominantly due to gardeners, but usually they're not doing it onto a scale that modern agricultural practices are. But this example was septic tanks in older homes that were leaking into a local estuary, leading these kind of blooms of algae. So the reality was, potentially, could you myceliate a biomass and act as a filter that it would selectively be able trying to survive, and basically exuding all these antimicrobial compounds to kind of keep things in balance. There's been a initial release for it done by Trad Cotter along with Daniel Reyes, and it's a bit finicky because the reality is that the fungus is trying to, once the fungus fully colonizes the biomass, it doesn't see the need to basically protect itself because it's established kind of this confluence. So it varies a bit. But this is kind of another example of that when working with fungi, you need to think outside the box and not just reduce them to being the sole entity. So microforestry is another thing too as well. The current way that we produce lumber in our civilization is incredibly destructive. Basically chop lots of ranges of forest down, and we harvest lumber, but we say, okay, yeah, but we do these practices where we plant trees. So we do these generational forests. So 50 years later, we'll come back and harvest as well. Well, that sounds all good, but the reality is, in general, is that one, all this biomass is left, everything is clear cut, and reality in general is that it's left there. It's not able to decompose and rot. It's not able to add organic soil matter content back to the soil. And secondly, as well, the majority of trees they actually planted these monocultures. So what's happening is that you're not trying to simulate the natural biodiversity that actually happens in these forest ecologies and habitats, but you're just trying to basically select species or strains of species that are viable economically, treating them as a crop, as a forest as well. And so the reality is, after second or third generation of 50 years, there's no topsoil left. So these trees get so big after a second or third generation that sometimes they just fall over because so much soil has been destroyed through the forestry practices which are extractive. So the reality in general is that, what's the solution? I don't know, mulch and compost. Sounds simple, like a no-brainer, but even if you took all that spent biomass and chipped it up and added it back, or even even if you just took 10% or 20%, which does not make sense economically, and just rather than actually cutting it down, you mulched and chipped it and simulate the natural kind of old growth succession that maybe the oldest trees that you lose to fungi that are any, maybe rather than incinerating them, letting them decompose, let successional biology do its thing. So it's a little silly, but the reality is you destroy ecology habitats, you get all this runoff. This is really bad, potential source for landslides, Pacific Northwest. And the reality in general is, why not just, compost, chip, and then really, the area is so devastated, you're not really worrying too much about, it's adding some biology back to basically help to bolster the natural ecology and environment. That's the whole situation overall. That's the whole aspect. So the reality in general is that you have the structure to soil that binds it, that helps build soil overall, and then consequently does not prevent this erosion. So micromediation, that's kind of where I got into about fungi. This was an image that I really like to show and talk about. This was from the workshop I mentioned where I learned about, applied mycology, all the different books on it. It's a piece of asphalt from the old Mosholu Parkway. The old Mosholu Parkway used to go through Van Cortlandt Park. And asphalt, it's made of 3 things. It's made of gravel, a binding agent, and bitumen, which is a type of basically petroleum product that actually is predominantly in the Canadian tar sands, but it's sticky, it binds together. And if anyone who's ridden their bike or driven a car or taken a walk on a street when after they just paved it, it's a sticky black tar when it's hot. So now once it sets and it's stable, it's great. I think a nice smooth road is kind of aesthetically pleasing, but when that road breaks down and gets all those little chips of the asphalt break off. So now these little bits of asphalt aren't just the gravel, but the binding agent and the bitumen are petroleum products that actually do contain a number of volatile organic compounds, polycyclic aromatic hydrocarbons. So here we see here a chunk of asphalt from the Mosholu Parkway that has oyster mushroom mycelium growing on it, and the oyster mushroom species from Van Cortlandt Park. This was something that was amazing about this. It was nuts. The fact is the fungus is basically adapted to basically find a way to extract the carbon, the carbon-rich structure, the hydrocarbons in the petroleum and binding agent to basically use it as a food source. This was really kind of blew my mind in general. And it's, that's something that really is striking. And it's, like I said, that's a non sequitur moment. So kind of focusing on how do we, compost spent biomass, building living soil, going back to the scary chemistry of cellulose and lignin, it's the structural bodies. And the reality is that the chemistry is related in that a lot of these polycyclic aromatic hydrocarbons are close to the building blocks of lignins. These phenylpropanes. So we call them phenylpropanes because there's a phenol group and they're propane. So it's the chemistry in general. But the reality is they're close to these cyclic poly meaning many, cyclic aromatic, polycyclic, poly meaning many, cyclic meaning circular, aromatic because they are aromatic compounds, and hydrocarbons because the aspect too is hydrogen and carbon. And the reality in general is that these are simplified diagrams, but these are studied with hydrogens. And the reality in general is that, volatile organic pollutants and then heavy metals are active because they can bind quite readily to our own biochemistry. So that's the main reason why they are a number of disease-causing agents and why pollution is hazardous to our health. And, in general, we think about plastics. Plastics are plastics are pretty complex in general. There's a whole range of organic chemistry dedicated to petrochemistry. And they are hydrocarbons, but very simple. But the reality in general is that one plastic product, which was one of the first plastic products, was cellulose acetate, which was pretty much in general, you take cellulose, like the same stuff we're wearing, and you acetylate it. An acetyl group is this structure here, COOH3, basically in general. It's bound to that as well. So the reality in general is you basically acetylate the cellulose, and it's stable, and also it's heat resistant, doesn't melt. So this is actually these were used to produce the first cigarette butt filters. When the tobacco industry realized that a number of studies are coming back that tobacco was carcinogenic and that smoking cigarettes was carcinogenic, this was the response. So the reality in general is that cellulose acetate does decompose. It's not an environmentally disastrous plastic, but it's the fact that you're loading, when you smoke a cigarette, the tar in the cigarette is full of these PEHs. You have a slurry of these these volatile organic compounds suspended in the sabot of the actual cigarette butt. So after cigarettes and smoke, it's flicked into the street. This arguably gets washed down the drain. And this is leaching PEHs into the water system. So it's a huge aspect. I was interested in doing these applications about how would you, look to break down contaminated biomass like petroleum and soil. This is Paul Stamets and Peter McCoy's work. Paul Stamets and Peter McCoy's on the right. The aspect that if you back mycelium, certain types of mycelium fungi, their mycelium into a corner enough, they will get resourceful. They will figure out ways to break down organic compounds they normally don't. So this was an example of, basically contaminating a bunch of straw that's been fully grown out with mycelium from an oyster mushroom and with basically petroleum hydrocarbons, and then the fungus, to survive, found a way to extract those nutrients. And then that was scaled up to a whole mound of oil-contaminated soil. So the middle images are from the Washington Highway Administration study that he did in general, which was really amazing because the aspect too is that you have a generational ecology in its ecosystem, but the reality in general is that, the volatile organic compounds, the heavy metals, they're actually being stored in the fruiting bodies. The mycelium doesn't want it in there. It's going to push them out. So the reality is, how do you contain the capacity of it? Maybe you could go on to compost them further. The reality in general is that good compost, diverse soil microbiology is incredibly active to do a whole number of biological processes that facilitates remediation. We'll kind of touch on that in a couple of slides as well. And on the right is a cigarette butt, which has an oyster mushroom fruiting off it that's been fully myceliated, contaminated cigarette butts. And that was kind of what I was interested in doing. So I was interested in looking at potentially different types of fungi that could do a similar effect. This is Cordyceps militaris. I think people have heard of cordyceps more recently, either from like Planet Earth documentaries of a zombie fungus, or from medicinal mushrooms in general, them being kind of a incredibly potent medicinal fungi. So this Cordyceps militaris, it grows in our region area, and I was thinking, okay, cool, like so insects are made of chitin. This fungus is pretty good at like infecting insects and then also being able to bore through, the chitinous outer shell. Fungi have chitin, but also so do crustaceans and insects, different arthropods. So I was thinking, okay, cool. Like there must be some way to basically understand that this chitin, this chitin molecule is. Correction, I should say what is it? I should say cellulose acetate and chitin instead of cellulose. Anyway, but this chitin molecule is far tougher than the cellulose and arguably the cellulose acetate. So maybe a better candidate for breaking down contaminated cigarette butts might be cordyceps. So this is a stack of mycelium, cordyceps mycelium, growing on a petri dish. It has this really kind of white, fluffy halo, and actually does start to pigment yellow after a while. This yellow and orange color. That due to a number of reasons, given the fact that I was teaching while trying to do this project, it didn't really take off too well. I do want to revisit it sometime because I think about there could be things to explore and understand that kind of systems approach and understanding the first principles of what enzymatic vocabulary certain types of fungi might have. So enzymatic combustion is the process by which fungi break down organic molecules. You have lignin modifying enzymes and cellulose modifying enzymes. Usually for bioremediation, you're focusing on the white rot fungi, the ones that actually break down the lignin, because they're tougher molecules and more closely related to petroleum hydrocarbons. So lactases, lignin peroxidase, manganese peroxidase, whole range. These really scary looking, organic, like words in organic chemistry, kind of generalizing the enzymatic reactions that do occur. The simplest thing you can talk about with, looking at these cool, aside from looking at these cool pasta-shaped little protein molecules of what they look like in reality, is the way they function is very much kind of like the game Breakout. If anyone who's old enough to remember it, or just that much of a nerd, with Breakout, you basically have a little paddle that releases a ball and will gradually break down this rainbow-colored layer of a wall, and the goal was to break out. So the reality in general is that you kind of have these reactive oxygen groups that are flying out. I think it's superoxides and other compounds that basically are just shredding apart the organic matter and allowing the fungus to gradually digest it. So that's a cool way. So yeah, like it's a cool way to kind of dive into that capacity, is understanding there's a lot more connection that goes on. I do want to dive into the fact that, when we think about environmental remediation, it's not just about fungi. It's not just about one type of organism. The fact that these organisms work very tight in unison together. This is an awesome paper from 2016. It's a review paper, so they basically take a number of studies from all over the world and talk about aspects, biological technologies for the remediation of co-contaminated soil. They're talking about that, and that basically soils that are contaminated, they're not just contaminated one thing. They often have a whole range of contamination and pollution going on, petroleum hydrocarbons, heavy metals, you name it. So the aspect too is they were looking at that most remediation techniques are usually predominantly focused on physiochemical technologies or biochemical technologies using different compounds to break it down. And the reality in general is like bringing in biology and a number of more emergent things are effective as well. For anyone who gets a little scared of genetic engineering, it's on there, and they talked about it as part of the whole aspect about there are some promising concepts, but the reality is you tend to have a DNR, a do not release policy, that it's worth exploring with potential down the road concepts, but oftentimes the current moment, far more effective means exist in the microbial remediation, or kind of using interesting structural chemistry using organic compounds or biological systems that are present already. So this is a great example, a great slide that kind of dives into really the whole process it's kind of busy, but it's showing that when you're having a mix of heavy metals and organic pollutants, what's happening with the microorganisms in the soil. So you have these bacteria and archaea that are actually living in these contaminated environments. They're actually probably, they've survived generations upon generations and have the genes that help them survive. And that oftentimes that might be the best area to prospect for what's out there. I know a number of people have looked into like maybe we should study the microbial populations that happen that exist in the Gowanus Canal or Newtown Creek or any other Superfund site in the United States, which we have thousands of, which is terrifying because we probably have microbial populations that are pretty well adapted. They're pretty tough. So it shows all the different potentials. You have co-precipitation, where an enzyme will tie up a range of heavy metals. You have the whole redox capacity, where you're basically taking basically a species of a heavy metal and going through a process of modifying its valence of electrons to make sure that it'll be less reactive or not as reactive. You have biosorption, so the aspect too where basically the molecule itself, where you have a organism putting out enzymes where the heavy metals are binding to, or it's doing the aspect of adsorption, where it's making this film on the outside of the microorganism. Absorption refers to bringing it into them. Adsorption refers to the surface. So this will happen for organic pollutants and heavy metals. Degradation, so actually what we'd like to have, or maybe some of these really tenacious microorganisms can actually use a range of enzymes, take organic pollutants in a complex with surfactants and other compounds to make metabolites and release carbon dioxide and water. And then you have the internal, when these compounds are absorbed as well. Biochar is really cool. We're still learning a lot about it. Probably biochar is in the same state that compost was back in the 1980s. Like when compost got popular, people were like, oh yeah, let's get compost, and like municipal systems would set up collect compost setups and collection, and people would get it and they put it on their plants, and all plants would die because people didn't understand that you need ratios of different organic matter, of carbon-rich matter versus nitrogen-rich matter, like browns and greens, and that if it's putrefying organic matter that's too rich in greens, it'll just kill your plants, and if it's too much brown, it just won't do anything. It just won't really decompose. So biochar is really cool. There's a whole range of situations. I'll just take a look at the paper. It's got pretty pictures. But this talked about how different types of biomass, based upon what material was sourced from, and what temperatures it was actually it was made or calcified or calcinated, had different effects. So we're actually at a part where we're just learning about that. Like biochar is pretty nuanced, that it's not like compost and biochar, all different types. It's a whole different world we're just starting to understand. So phytoremediation. So phytoremediation usually is a lot of people's introductions into bioremediation. A lot of people are like, oh yeah, lead's like plants pull up all this lead and we'll just take them out and pull them out and we'll just do this and do that. And it's a little more complex because, different species of plants do different things, and also a whole different range of situations. Most people talk about phytoextraction, where plants uptake these materials and they pretty much will bind them into their they'll basically bind the heavy metals into their lignin molecules, or they'll basically degrade them and then make metabolites. However, there's interactions with microbes in the root system that do a lot of process I showed earlier. There's stabilization when they put them in lattices. And then one thing they don't really tell you about is phytovolatilization, where the organic compounds might be broken down, the inorganic compounds can actually be passed through cell walls. Maybe they'll be stored in vacuoles, but maybe complexed with like a ligand, like they'll have this formation and stored in a vacuole, but also could actually be respired into the air, so they can be aerosolized. So phytovolatilization. So there's a thing that with phytoremediation to be mindful of, and it's just a whole situation. So the effective plant microbiations, that's important too. You can't just say one plant, one situation, they're all over the place. That's basically the big thing in general. Lots to learn. Cool. And this is the aspect too, how do we work with them? So these are the really kind of cool, like electrokinetic techniques. It's really cool. Like they talked about how people literally put electric field in a contaminated area to try to like change the electrical conformation of pollutants to make them more available. It's pretty wild. There's all this stuff, but showing that you're using surfactants, chelators, organic acids. This is like for stuff that's just been like destroyed. Like it's there are professional environmental remediation firms that have all these different techniques. But the aspect too, how do you integrate after it's been washed or electrokinetically treated with biochar and compost? That way you move it more towards something that actually would support life. So cool. And then if you really want to nerd out like and do math, this paper came out I think in 2019. It talked about the biological presence. It really kind of dives into like multi-metal removal. Yeah, so if you want to check that out as well. Spontaneous multi-metal removal from soil with washing agents of waste, plant, and microbial origin. A bunch of scientists based out of Poland. Pretty neat. So how's everyone doing? Okay, we got some questions. Looks like it. So I'm gonna maybe we'll just take a minute to answer these. Cool. Let's see here. Where do we start? Okay. So Jody, do mushrooms create new hyphae from the spores dropped by fruiting bodies? Does the newly formed hyphae join into the existing mycelial network back in the soil? So if I don't know the answer to that question, it's easy to say yes, but I don't know if that's 100% true. I was thinking about the factors that, given the fact they're from the parents, and they're recombining and then ensuring that, well, they recombine. Possibly, not 100% sure. I'll have to look that up, but it's a great question to dive into. Let's see here. Oh, like soup to inoculate coffee. I could do a video on that, but the easiest way to do that is like literally just take some cardboard, get a nice little trough, make sure the cardboard doesn't have any like tape on it. If you want to grow it to eat, if you want to use it to make mushrooms to eat, maybe don't use any cardboard that has any ink on it, or if you're just going to mess around and experiment, feel free to have it. Make sure there's no tape on it. Get some boiling water. You can pasteurize it, pour into the water. The hot water will kill most of the microorganisms that might compete for it. But also you're going to be hydrating the cardboard, and also you'll heat up. You'll basically dissolve the cornstarch-based glue that holds the corrugation to the paperboard together. And whatchamacallit? Yeah, and so you'll allow prep for inoculation, and so it depending upon where you're living with your tap water. Most tap water in the U.S. is chlorinated. Chlorine is put in it to kill pathogenic microorganisms, which fungi is a microorganism as much as it can present itself macroscopically as well. So if you have just chlorine, you can stir up the water, leave it out overnight. If you live in like New York City or the New York metro area like I do, we tend to use chloramine, which is a bit more stable. If you're worried about it, maybe just use some spring water. Spring water will have a bunch of trace elements in there as well. Distilled, maybe not so much because it doesn't have trace elements. And then just you kind of heat it up, separate, and then inoculate it, and then put into a plastic bag, sandwich bag, seal it, open a little mouth hole, roll that up, and then put that into a brown paper bag. The plastic bag retains moisture, helps keep it warm, and allow passive kind of air exchange. And then the brown paper bag kind of keeps it dark. You want to simulate this environment that's like in the soil or inside of a log for incubation. Happy to point you to resources. Compost from local mushroom farm. What should I do with it before using my garden? Okay. So what should I do with it before using it in my garden, or it is ready to use right? Yeah, just break that stuff up, man. Like honestly, it's a great mulch. Like if you have like regular wood chips as mulch, like just literally like throw it down, layer it. It's great. You can throw it straight on. Just if you're gonna throw it straight on, break it up finely. And if you have access to mulch, I think it's great if you break it up and then like wood chip mulch, put it on top and just water it. That helps to prevent the mycelium from drying out, and then also the mycelium will grow up into the wood chips. You know, if they're the mushroom farm, they're probably growing like shiitakes or oysters, which are the primary decomposers as well, for sure. And then even like, you know, like compost living species, like it's just good compost, dude. Best way to grow a thick layer of mycelium and dry for textile applications, similar mycoleather. So yeah, the Facebook group is great. What Ecovative does is, and you know, just from how I look at their system, is, you know, this, I don't work with them, I just look at how they do it. They have these like big troughs, like these big, they almost look like fish tubs for food service, and they pack it full of mycelium and they throw it into a container where they probably pump in CO2. Now fungi, they breathe like we do, so like they don't like hard carbon dioxide environments. If there's a lot of carbon dioxide, they'll grow up out of it, and they're probably putting on a substrate that like doesn't is deficient in nitrogen, so it's not going to produce fruiting bodies. So the mycelium is like, get me out of here. So it makes this long, like stretchy thing as well. But yeah, there's a lot of cool ways to experiment. I think a lot of people are doing some crazy, awesome stuff. Like Phil Ross is doing some cool stuff with MicroWorks, with the reishi leather, which is pretty neat. So, and there's so many damn species of fungi to play around with. It's for sure cool. All right, so that's the question section. So I guess we'll get back to beneficial microbiology and soil and aquatic ecosystems. Oh, yeah, so we talked about the soil food web. You know, we tend to think of ecologies and environments, land and water as two separate ones, but the reality in general is, if anyone's ever kind of you know, looked at evolutionary biology, like from when understanding that life started in the sea and then moved on to land. You know, life's pretty good in the sea. One thing about permaculture is that you want to try to facilitate environments where there is access to water, and which has a lot of insights to the kind of the hydrogeological chemical system as well, which we'll talk about with Sergei Winogradsky too. Anyway, but yeah, food soil web, that's important. Bad soil, no structure, lots of farm particulate matter, lots of bacteria, very little organic matter. Fungi, they break it down. Remember this slide, structure, making these macroaggregates, they're pulling all the microaggregates together. So a big part of this is the carbon cycle we just talked about, but the nitrogen cycle is hugely important. You know, the fact that most of our atmosphere is nitrogen, but we can't drink it. Can't drink it. We can't, like, well, life doesn't utilize it. If you any bit of nitrogen that you had to build our bodies, it came from the fact that basically a plant, microorganism, or some relation between the two were able to fix it. And it's the reality in general is you have these nitrogen fixing bacteria in soils, and those basically go on to basically provide the building blocks for these proteins and organic acids. Bacteria and fungi decompose them. Predators, protozoa, nematodes eat the bacteria and fungi. They poop out plant available nitrogen, and obviously, like any kind of plant root feeding microorganisms or predators will actually poop out that from plants. Then the concept of mineralization, you know, you talk about, you know, with perennials, you know, the factor in general is going to be returning this capacity as war. And then going through the nitrifying bacteria process, we go through the aspect where you have basically brain fart for a minute. You have ammonium, which is NH4, going through the process of nitrification, getting oxidized, it's getting made to nitrite and nitrate. You need that for annuals. Perennials, they require ammonium as their NH4 plus. That's why you tend to have, for like shrubs or trees, they're more fungally dominant acidic soils. When we say fungally dominant, it's just there's lots of fungi. They mediate the relations. They're not the only thing living there. They actually make lots of structure and ecologies for other microorganisms. And then for your annuals, like your vegetable plants, which are like high energy plants, they require more nitrite and nitrate. And which is why, like in a lot of urban soils where there's tons of bacteria that can nitrify, that basically are symbionts. They're symbionts with a lot of these weeds like dandelions, mugwort, Atlantis, tree of heaven, anything that's growing up out of an urban sidewalk crack. It's like, how does that plant do that? Because it's microbial symbionts are predominantly bacterial, because it's an annual of the most annual variety. Cool. So here's a cool little diagram. Matt Powers, really awesome. Check him out. Really awesome permaculture educator, really accessible. He's got he was a high school teacher before, has his master in education. He has this amazing diagram, which kind of talked about the kind of this yin and yang, and as much as new agey that may seem, but or even depending on perspective of a foundational concept of your culture's theology and philosophy. It's kind of this balancing effect back and forth. And that bacteria thrive in these alkaline environments, and they produce a lot of these nitrates. They're basically doing that, and then basically fungi facilitate these other conditions as well. So the reality in general is that we're learning a lot about how important soil biology and microbes are to our health. We're actually realizing, hey, maybe like the fact we're living more and more like in urban environments or artificial environments is a way different than the environments our ancestors grew up in, right? Like maybe like our microbiome is part from like our parents, like your mother gives birth to you, like unless you were birthed through a cesarean. Basically in general, when you're born, you are inoculated with microbiology from your mother's vaginal canal. And then even too, when you're, we're learning even too that you're getting that as well when you're breastfeeding. And then also to environmental. So the source pool is this primary inoculation. So that's how the host selection actually works. And even more so, we're realizing it's, soil is largely contributing to the soil microbiome, and how the factor in general is that we're kind of slowly peeling away that, that a lot of our ancestors were living lifestyles which were much more directly connected with ecology and nature. Like often things that were waste weren't volatile or hazardous because, they weren't an artificial concept that it was highly processed or manufactured, that where a lot of the waste products or byproducts might be volatile or pathogenic or just reactive from a chemical standpoint. Mind you, there's plenty of stuff that naturally occurs in nature that can and will hurt you and evolve that way to do so. But, from a casual thing that often is quote unquote a waste product, it can readily decompose. So we're kind of learning more. And then how, as we move more into these artificial environments, you have a situation where different inputs, part of our growing food and how they're stored and how they're preserved and how they're treated can affect these aspects. And the reality is microbes, they're going to survive because their reproductive cycles are way faster than us. So, we're learning that a lot of humans, especially living in developed environments, we are a predominant key role player in our environment are bacteroides. There are species that kind of the cosmopolitan contemporary bacteria and human microbes, and that our ancestors and even people that are living in suburbs, or even living in rural areas, or even living on farms, have a much more diverse and complex microbiology. And we're kind of starting to realize there might be something to the effect of how these microbes help kind of relate, and that it's not just I got microbes, like they're germs, kill them. No, it's like you are just as many cells that are in your body that are you, which are 34 to 35 trillion with a capital T. Like, let's talk about perspective of scale for numbers. Like a million seconds is like just over like 2 weeks, like 17 days. A billion seconds is 31 years. A trillion with a T, trillion seconds is 31,000 years. So you have roughly 34 to 35 trillion cells, and there are just as many that are microorganisms with 100 times the species variety and 1,000 times as many genes. And you look like a person, you look like an animal, you look like a human because your cells are 100 times bigger. The factor is these organisms are living on, in, and around you in every single crack, crevice, orifice, and roughly interstitial space possible, because you are a natural ecology and environment as well. So its reality is like, it's to think about it is that, really it's literally your second organ system. And we're even think about this is like for cell biology, we tend to forget that we're a human, we have this very idiomatic experience, but the reality in general is your 35 trillion cells of different types that are expressing set parts of your genome, and they're expressing them to achieve a larger coordinated function. but the reality in general, there is variety. It's an ecosystem collaborating. It's just a more so of how much that cell pools from its own genome set of different proportions that it's expressing, but also silencing. Silencing is also very important. You wouldn't want your eye cells in your eye, like a rod or a cone, which is a photoreceptor. And then you have in your gut, you have epithelial cells. You have cells that are actually produce digestive acids and enzymes. You wouldn't want one of those digestive cells in your gut to start acting like a photoreceptor in your eye. Or vice versa. You wouldn't want the eye cells are acting like digestive cells. So, expression of genes to what they become is just important as silencing or suppression. So, but the reality in general is that think about how much those interactions of genes are working with one another in your own cells, what interactions are happening with other microorganisms in unison. So, yeah, that's a big spoonful. So then also we're learning there's endophytes in plants. So microbes don't just happen on roots, that they're also in plants, but also in seeds, vertically transmitted endophytic, basically endophytes. The reality in general is that when a seed will germinate, I'll read this little quote. The relationship between endophytes and plants is unique in the ability of the former to provide alternative sources of biologically active metabolites such as enzymes, biofunctional chemicals, phytohormones, nutrients, and minerals to facilitate the distribution or production of these resources, which contributed in the elimination of various stresses. In return, the host plant provides a protective sanctuary for the microbes within the plant tissues, in which they can grow and reproduce, but without compromising the plant's own growth resources. Yeah, it's the fact is symbiosis is literally the way that plants moved on to the earth, onto land. It's the fact is that, nothing lives in a vacuum. It's 1 + 1 = 1. It's the fact is we're thriving with biology. And, a lot of it is we probably only realizing this now because a lot of the process by which we acquire information is reductionism, which isn't to say it isn't a bad thing or against it, so that, we've gained so much knowledge through this process of reduction, but we're realizing that, wow, there's a lot of intersections. The top-down approach is effective at really distilling and pulling things down. And people do amazing graduate and doctorate work refining and pushing the limit of human knowledge, but we're starting to realize that the bottom-up approach is just as important, and that we're able to distill what we've gotten top-down from bottom-up and look at all these interconnections. So this is an example of talking about the indirect versus direct systems, about how the symbiosis benefits isolation and maybe try to apply them versus stuff's in the soil. It's probably these plants are surviving in these conditions for a reason. Let's try to just really just push biomimicry as much as possible. And then we have the concept of vertical transmission. We're actually realizing that a lot of these endophytes, you basically have the aspect where these environments where microbes listed, it's the roots, it's the rhizosphere, the phyllosphere, you have the germosphere, and then even too you have the fact that they're transmitting into the seeds, that potentially that some of these microbes are critical for seed germination. They're literally microbial triggers to basically jumpstart it. And then even too with fungi, you have endophytic fungi. You have mycorrhizal fungi that grow either outside of the plant root or between the plant root, or even make an arbuscule that make a structure that are a mycorrhizal symbiosis where you have an imagination of plants to exchange nutrients. But then you have endophytic fungi, which is the fact that you have some fungi that have lost the ability to even produce spores. They're these ancient fungi that are just living between this vertical transmission. And dark septic endophytes are one thing. We thought, oh yeah, you can count more plants. It's easier to count the plants that don't have mycorrhizal symbiosis than do. We previously said, 90% of plant families, 80% of plants. But then we found out about dark septic endophytes, oh yeah, it's all plants do. And then we're realizing it's not just fungi, it's everything. It's pretty wild. And then we're learning about stuff like rhizophagy. So out of Rutgers, James White has done a lot of amazing work on this. Elaine Ingham's work talked more so about the role that the predators do in basically just, eating the bacteria and fungi that have the solo reserves. But rhizophagy is the full-on example of where literally you have a proof where the plant is disabling an enzymatic capacity, the superoxides that the bacteria might produce is actually dissolving its cell wall and intercalating it and extracting the nutrients and spitting it back out. This is like crazy. It's like, this is basically turning it's like, it's thinking about like how Jethro Tull was like, oh yeah, like let me invent rototilling because let me break up the soil because like plants have little mouths. This is kind of like an interesting thing. Like rototilling in good capacity works one part, but like, it's pretty crazy to go from like Jethro Tull to like rhizophagy. Like that's pretty wild. And then, we talk about aquatic systems, right? You think, oh yeah, like aquaponics, aquaponics came out of the from an aspect of aquaculture, like, like, really our practices on producing food, are destructive from like overfishing. And so how do we grow food? How do we grow fish in containers? And so like this happened back in the 70s. They started growing fish in these isolated systems. They found that there was such a huge problem with disease that literally there was so much ammonia being produced from the fish waste that it was causing pathogens in the fish. They were like, okay, cool. Maybe we should just grow plants. Oh, if you grow plants with the fish in these tanks, it actually helps. And so it turns out you're like, oh, well, duh, you're mimicking a natural riparian ecology. A riparian ecology is where like land meets water. We know that in permaculture, like it's this edge effect. And permaculture is that understanding that you have a select set of organisms that have life cycles on land. You have a select set of organisms that have life cycles in water. What happens when land meets water? And you have these gradual kind of cycles of time over between like what happens over the course of a year. There are organisms that have a doubling effect where you have species that's actually spend part of their life cycle in the water and then move on to land and vice versa. So it's this whole aspect where like, wow, like with aquaponics was like, you're really trying to mimic a natural riparian ecology, and how it's pretty amazing because you do have these nitrosomonas and these that are basically these ammonia oxidizing bacteria making nitrates, and then nitrosphere turn that into nitrates, and then feeding it, and it's a whole capacity overall. And then the reality in general is like this is what happens in natural riparian ecologies. Like, and then so I think in general there's a lot of potential for bioremediation, like that. Like we understand that the Newtown Creek Alliance, which is an amazing community conservation community environmental conservation organization, they did these things called living docks, where they literally made these they took these pontoons where they plant these saltwater marsh grasses in the Newtown Creek. And we have really brackish waters in New York City because we only not only have the edge effect of land meeting water in our estuaries in New York City with all our islands and all our inlets and channels, but also the fact you have brackish water where fresh water meets salt water. So now you have a quadrupling effect of species and biodiversity. So the fact is that everything is going on in a cycle because it's this larger symbiosis and this scales. As much as it is macroorganisms, microorganisms, it's the factor is that it's these nesting symbioses that go on from a small scale to a large scale. So this is another example. Matt Powers did this diagram. He does great diagrams. Can't plug Matt enough of explaining the details of the nitrogen cycle in aquatic. But I want to pay attention down here. What happens in the sediment, right? We talked about what's going on in water and soil. Like, what about in the compaction layers? What's going on? Well, like, we know the nitrogen cycle does happen in that capacity, but so this is talked more about aquaponics. This is a different presentation. Just talked about more the aquaponic research about how with recirculating aquaculture, it's better just with plants because microbes show up and all that kind of stuff. But yeah, so here's a great example of what happens in that kind of that lower oxygen layer down here. Well, you have a zonation effect. So here's a Winogradsky column. So Winogradsky column is a cool way of where anyone can do this. It's a great high school activity. It also talks about how do you Winogradsky was a contemporary of Pasteur, Tyndall, and Koch, who basically, who Petrie was worked with in general about, whereas they were all studying in general, they were studying microbiology, but they were looking more in isolation. And so, Pasteur and Koch were studying more pathogenic microbes, so it was essential for isolating. So when Sergei Winogradsky was like, wait, what if we look at the whole environment? Where we understand that all these areas, they have different conditions where this is a dynamic system. It's not static. So you have a gradient. So we have a Winogradsky column. So Sergei Winogradsky, it's really amazing now because it's a cool way how you can look at these snapshots and profiles. This is anything but a static environment that does not live in isolation. These are incredibly complex systems. So to gradients and zonation. And you're seeing that these different areas where you have an oxic zone where cyanobacteria are living, where it's like the bottom layer, it's the top of this, it's basically the bottom of this pond and the top layer of this area, where you have light and air. So you have cyanobacteria, and you have the different basically conditions. You have photolithoautotrophs based upon where they get their energy, where they get their electrons, where they get their carbon. So they're phototrophs, meaning they use the sun. They're photo, meaning the sun. Litho, meaning that they're actually extracting their electrons from actual inorganic material. And then autotrophs, which means that they're basically producing their own food. They're basically they're pulling carbon out of the diffused in the environment and producing that as well. So they're basically pulling in carbon dioxide, producing organic compounds. The example there is ammonia, or the whole range of organic as well. And then also too, they're basically pulling in water and producing oxygen. Heterotrophic bacteria, they're going the whole process where they're basically taking in those byproducts that are produced from the cyanobacteria. They're making carbon dioxide, and they're pulling in oxygen and producing CO2. So they're chemo-organo-heterotrophs, and the whole thing goes on. And I can link you to an amazing diagram from the Howard Hughes Medical Institute. So pretty neat in general. So yeah, there's a whole range of how this stuff works out. It's pretty amazing. But yeah, but the reality in general is that it shows that like life isn't static. It's incredibly dynamic and interconnected. And these are really amazing, because when you do these columns, depending when you seal them or keep them open, you'll see these different blooms of microbes happen overall. So it's amazing how you could understand these environments. Whereas in the soil, it's static, but aquatic systems are incredibly dynamic. So yeah, come back to fish. Really important. Cool. So we talk about microplastics are a huge thing in our environment, right? Like they're coming off our clothing, they're coming off the sea is full of plastic. Oh my God, it's terrible. Well, here's the thing about it. Like the fact is that they're one range of fungi that we haven't talked about are aquatic fungi. We talked about a bit about them in the last couple of slides, but in kind of vague connotations. But we've only studied terrestrial fungi because we live on land, right? Aquatic ecosystems are incredibly more diverse. And so this basically showing that potentially microplastics are altering these compositions of fungal communities in aquatic ecosystems. How they're actually making biofilms on these microplastics. So it's really amazing that these might be adaptive overall. So I'll let you guys dive into that paper. And then even to like different eukaryotic life on microplastics and brackish ecosystems, like an ecosystem that happens in New York City, where like you have a meeting of fresh water and salt water. And yeah, it's just so many black holes to dive into. Fungal foods and medicines. For the 50 of y'all that hung around, we'll kind of dive into this more of the kind of traditional, more practical details of the talk. Growing mushrooms, food and medicine. Remember that. Mushrooms are an excellent source of protein. 20 to 30% protein by weight for most of the common cultivars. They're mostly water, so you don't got to add a lot of oil. America's Test Kitchen has an amazing video how to cook mushrooms, explaining that they don't treat them like a vegetable. They're actually tougher to cook. They're actually tougher to overcook than meat and vegetables. But yeah, they're basically in general they're the cornerstone of civilization. Every single food, they're playing a role in fermentation in some capacity of a symbiosis of extracting nutrients. From baking bread to our sourdough starters for fermentation, the list can go on. Also, like booze, come on, like civilization. I think the whole beer before bread thing makes a lot of sense. Why would humans go to all that trouble to grow all those grains? Because fermentation is the bomb, rather than bread's a nice thing as well. But I think the start of the agricultural revolution for humans was probably a bit more bleak. So whatever kind of cured that cured that ailed back then, but in general. But yeah, microorganisms, Saccharomyces cerevisiae, star of the show, and there's a whole aspect of how we've developed certain strains and cultivars of yeast that do all different sorts of things, and even to the provenances. The reason why like champagne wine is called champagne wine, because the yeast that show up there produce that range of extra carbon dioxide. So it's amazing understanding like a terroir based upon the conditions that lead to a provenance, and getting into food stuff as well. So, so profiles of medicinal fungi. What mechanisms of biochemical action are behind medicinal fungi? So this is a nice little diagram. This is pretty neat because like You know, the thing about it is that, you know, we're at a point now where we're learning more and more about like the chemistry that goes on between plants and fungi, and then, you know, why we are selecting these for different applications of food and herbal medicine and details like that. Um, you know, I don't produce or sell any compounds or products. I'm not a medical licensed professional. I'm just articulating sciences out there. There's an incredible amount of research going on. There's a lot of amazing integrative institutions. Sloan Memorial Kettering Cancer Center has a great integrated medicine website which does all these reviews. But the general breakdown of the theory about what's actually happening is that when you're when you have these plant or mushroom products that you're processing, you're basically planning to extract a range of different compounds, either hydrophilic compounds, which like water, which means you can extract them with water. You're basically making a decoction. It's a fancy word for a tea. Polysaccharides, or you're making a hydrophobic. You're extracting the hydrophobic compounds. You're usually using some kind of non-polar solvent. You're using an ethanol extraction or some other solvent in general, and often you're going over these sterile molecules, these flavonoids, these terpenoids. So it's understanding that these hydrophilic compounds are immunomodulatory, that they upregulate the immune system. For speaking of the studies, you can just follow John's link. There's so much literature out there, and it's going in a cool direction now. The biggest thing with studies is how big the sample size is. It's about when you see a study in a paper, you understand that you have to look at the methodology of how experiment was performed, because just because you see a study doesn't mean it's effective. So you have to use a little bit of a discerning eye. But yeah, the one John linked in the chat is a great way to look into it. But yeah, and then the aspects too with ethanol or basically solvents, because water is a solvent, but it's a polar solvent, versus non-polar solvents used to extract these sterile compounds. So the aspect too is that these understandably slightly suppress the immune system or regulate it down, which helps for inflammation or reactives. And then even understanding if you have a good microbiota, healthy gut microbiome, or just a microbiome in general, that these polysaccharides, these hydrophilic compounds, one example is polysaccharides, actually get digested by these short-chain fatty acids, will actually get digested into short-chain fatty acids, and then basically provide a downregulation, immunomodulatory effect as well. So it's a whole wrinkles and wrinkles of, it's amazing. It's kind of crazy. So the one that everyone kind of raves about, are these alpha and beta glucans. So these are the structural sugars that actually hold up the cell wall. These are these huge triple helical sugar structures, and this bracketed thing is a monomer. It's like one unit. You'll have billions of these, absurdly large amount. And the reality in general is these are big enough in macromolecules. It's thought that they basically interact with our immune cell types to stimulate an immune response. And it goes off the concept that since we're more closely related to fungi evolutionarily, a lot of these receptor types are baked in to our own kind of immune systems. And it kind of makes sense. It makes sense in a way that these are holding up the cell wall. Basically, they're between the pellicle, the manoprotein, glycoprotein outer layer, and then the chitin. So it makes sense if you're a fungus, you're made of hyphae, trillions of hyphae, you're one cell wall thick, and then basically you get punctured. Makes sense that these molecules swing into your cytoplasm. It's a very effective way for triggering an immune cascade. That's the way I kind of think about it in general. So they're enormous, and these are water soluble because they are polysaccharides, meaning many, poly meaning many, and saccharide meaning sugar. Hydrophobic terpenoids, sterols. So these are one example of the hydrophobic compounds. So these are a huge aspect. Basically, like these compounds in general play a huge range in these conditions and signaling in inflammation regulatory systems in our body as part of our endocrine system. And so the one we see here on the right is betulin. So betulin, if anyone is a fan of chaga, or is a keen on botany, betulin. Basically, betulin leads its name to it's basically the plant, the plant it's sourced from, birch trees, the genus anyway, tongue-tied. But the reality in general is that so we understand that in Eastern European and Slavic cultures, that birch sap and syrup was prized as an actual as a folk medicinal herbal kind of use in general, because it's the aspect when actually rendered in basically when rendered properly, it'll basically be turned into betulinic acid, which is a rather potent antioxidant. And antioxidants in general are intentionally kind of taking these reactive oxygen groups, these quote unquote free radicals that prevent oxidation. So they're antioxidants. You know, it's understanding that, you know, beyond the buzzwords, there is actually a bit of chemistry going on. They're basically helping to alleviate this this metabolic stress that's actually happening in this capacity as well. But lots and lots of details. Honestly, like Olga, I think a lot of people freak out about what's the best way to do it. I think in reality, it's just the factor is if you're getting something, it's good. You know, I think there's a lot of research that needs to be done to like evaluate concepts, but I like the tea a lot. People make a number of amazing, great extracts. I would say the one thing is maybe opt for, you know, this is not medical advice, but I would think about with food. I think about with microbiology that maybe sourcing, you know, plants or fungi, whether it's food or medicine, with nutraceuticals in that capacity, maybe from your own bioregion, in the area you are, because the climate and conditions that you're living in are very similar to the ones that have complementary microbiology in that way. So it's a larger statement to say, but it's from a conceptual standpoint, that's how I would articulate it. Let's see here. So, and also too, FIPs, fungal immunomodulatory proteins. So these are really neat because, so like, you know, antibodies, they're pretty important. They basically help to like stick to pathogens and tag them for our immune system to go to work. And, you know, fungi also have antibodies. These are basically fungal antibodies, fungal immunomodulatory proteins. It's the reality in general is because we're both epistoconts and we have comments from 1 billion years ago, that a lot of the similar structures, a lot of these fungal antibodies, FIPs, have similar basically structural chemistry, these proteins and how they're formed, and also can have complementary effects and how they're set up. This is really emergent. The reality in general, this is probably why eating mushrooms fresh is the best one, and also just incredibly good for you because they're stable. You haven't denatured proteins like the beta glucans and like the sterols. They're pretty stable. Like they're chemicals, like they're compounds. But with the proteins, that they're made of amino acids, so they do degrade over time. You know, so it's the reason why things, yeah, that's basically in that capacity. That's another thing. Some people do this crazy thing called mushroom juicing, where it's like you take like fresh mushrooms, you take the mycelium that's grown from them, like probably off a grain that's like edible. You throw them in a blender with like some kind of oil, and when we say oil, we mean like a food grade oil, like olive oil or something, and then with water and blend them up. You're basically making a lipid encapsulation, and then they would basically pour this through a nut milk bag and strain it, and then drink this slurry, which would probably taste gnarly, but it's like the aspect too is you potentially would be getting a fair amount of the biologically active compounds that do degrade pretty rapidly. So cool. Oyster mushroom species, oysters. They're great. They grow themselves. You know, they come in all different colors. They're good for you. So it's bye, Jenna. Have a good night. Yeah. So basically, in general, what is it going to talk about? Yeah, so yeah, they come in all different colors and, you know, anyway, but yeah, so they're pretty prolific, and we actually think they're not actually like subspecies. They're actually this large cluster because they adapt in different environments. So those pink oysters, those are Pleurotus jamor. They actually originated, I think, from Southeast Asia, so they grow really well in hot temperature climates. The yellow oysters grow in pretty temperate climates as well. I forget the species name for that one. I forget the Latin name for that yellow oyster. And then the blue oyster, Pleurotus. Well, there's a couple different types. There's just a bunch of different oysters, but the reality in general is they've adapted to different. They've adapted to different types of temperatures and conditions, and loaded in vitamins D D2 and D3, you can prepare them in a certain way where, much like how we basically absorb, basically in response to UV radiation, we upregulate vitamin D, which is why you need sunlight, which is why we need it, as well as that potentially mushrooms also do that as well, which is a source that they can, if they're grown outdoors or they're harvested and basically dried in the sun. So it's pretty interesting, and the exponentially large amount of international units of D vitamins that could be increased. And they're loaded with amino acids, and they just taste good. They taste incredible. And you grow them year round because you can kind of do this kind of stacking functions inside real timing with which temperatures they grow based upon whether you're living in a temperate climate or that climate that has a pretty big shift in temperature from seasons. Shiitakes, probably my favorite. Lechonoiotes. We've been probably cultivating them as a species. We've probably been cultivating them as a species for over 1,000 years. Like, and whoever's had like shiitake stem butts, like they're pretty gnarly. I like to imagine like maybe what was the first shiitake like? It's been like one gnarly mushroom that someone probably was like, "There's something here to work with," and here we are today. But yeah, just they're wood decomposing fungi. They're crazy to grow. They make a skin on like a substrate block if you try to grow them on sawdust. Most people grow them on logs because they're really effective, and they're basically super tasty as well. So when we say different types of beta-glucans, lecithin, so a number of polysaccharides are named with respect to the namesake of the species they come from. Number of studies based upon how they could. One example is B lymphocyte production. You could dive into all the different research that's been done, which is good. I think people should dive into the research because it helps them understand like how do you look at studies critically and understand the conditions and methodologies that make them actually worthwhile to articulate. Lion's mane probably it's having a moment right now. Delicious mushroom, looks crazy. Looks like a my friend Rich said it like told me I was like, I saw this mushroom when I was hiking up in the woods, and like, could you describe it to me? He's like, he was pulling out his phone. He's like, yeah, man, it was like a hairy fortress. And I'm like, okay, yeah, and it's funny, like, you tend to have with mushrooms with describing them these adjectives smashed through their nouns in the most weird ways. Non sequiturs, yeah, but delicious. The star of the show with these is that they tend to have a number of compounds, a couple different species of Hericium, Arsenines and Hericicones, basically these terpenoids and phenols that are small enough molecules that potentially can cross the blood-brain barrier and stimulate NGF, nerve growth factor. So there's a number of anecdote and a lot of techniques for them as well. They taste amazing. I have a friend that makes like vegan crab cakes out of them, and they slap. I'm just going to use that meme. They're just so good. And yeah, so there's a lot of good stuff as well, and they're just kind of fun to look at. The little pom-pom mushroom. Reishi, my friend Olga took this picture. Olga told us her birthday was yesterday. Runs Smugtown Mushroom. Definitely check her out as well. John's in the chat too. He runs Catskill Fungi. A lot of awesome. The myco community is very much alive on Instagram. Really love it. It's really great, especially having the Northeast as well. Reishi, Lingzhi, Ganoderma. There's a ton of Ganoderma species. Kind of goes in the whole thing with like polypores, and Paul Stamets talked a lot about like how benefits of polypore mushrooms, and it's looking like polypores are pretty effective overall. It's smugtownmushrooms.com. So Smugtown is the namesake of Rochester, and then I'm not sure if John's still in the chat, but it's catskillfungi.com. So John Michelotti, Olga Tolgas, Tzolgas. Cool. Someone can link it. There we go. Cool. So yeah, so urban mortality, like traditional Chinese medicine, they have a lot of medica materias. It's really revered. So apparently looking at, there's so many different species, and apparently they're loaded with these sterols, a whole range. They call them ganodermic acids, and this might be the reason why they're so effective, is because they'll help down-regulate inflammation in a whole different numbers of ways. The big namesake people talk about is that it stimulates a natural repair pathway called TP53. So it's tumor protector 53. It's basically a cellular tumor antigen. So it's basically it's called TP53 because they're 53 kilodaltons, this big complex that goes around and just kind of like error reads your DNA, and potentially in reality tumors are happening because something has happened to the genetic information of a cell. So we have a set number of genes, they're called oncogenes, which can potentially lead to carcinomas. I'm not a doctor. I'm just from research I understand and articulate it. I speak in general terms to inform people to do their own research and read with these aspects. But this is the reason why it's gotten a lot of attention for clinical studies and details. Also just looks beautiful. Cordyceps militaris. So cordyceps, people are kind of blown up. People have heard about it. Like this insect fungus. It's got a lot of infamous and famous aspect for athletic performance and libido. We'll leave it at that. But it's basically, in general, Cordyceps sinensis is like the Tibetan caterpillar fungus. It's problematic because people will die in the kind of trafficking of it because it's so valuable, because these fungi only grow on the Tibetan plateau because these organisms have adapted to the biotic conditions and biotic and abiotic conditions that are high altitude in this kind of cycle with certain types of insects. But however, Cordyceps militaris grow predominantly here in North America, and they're really interesting because they're yellow, they're crazy, and they're a potent pathogen for intimate pathogenic fungus. So yeah, there's all different sorts of stuff. Apparently, they produce this compound called cordycepin, which is an analog for adenosine, which it's thought to basically be a template for making more ATP, so helping drive a cellular metabolism. Why would a fungus do this? Well, most life needs ATP to run. That's why eukaryotic organisms have mitochondria in the first place. But it's the thought that if I'm a fungus that's going to parasitize a insect, a pupa, and I want to keep it going up until it fully is mycelium, I would literally start churning out a precursor to a compound that would literally drive its metabolism. So that's one thought I had. But there's a lot of potential as well. Yep, and also so mycosymbiotics. So that top picture is from William Pydia Brown's run. Good friends with them. There's Cassandra Posey. She's actually not painted orange. That's just the glow from the image. It's pretty crazy looking. So they run mycosymbiotics, and Cassandra. Well, mycosymbiotics is William's effort, and then Cassandra runs cognitive function, which she does a lot of products, and you can grow them. Yeah, grow them in jars on rice, and it's pretty neat because you don't need to have like a special fruiting chamber. They fruit in the jar, so it's pretty amazing. And William does a lot of amazing work with going out and prospecting those strains and breeding and doing single spore ascospore isolates, which is pretty awesome. He also really caught the molecular biology bug and started doing looking for presence of mating types, MAT1, MAT2, which is apparently an Ascomycota mating type, so determining what spore is compatible with what other spore. Well, so to clarify, Sally, Ophiocordyceps sinensis, or the black cat, it's the black fungus that comes from Tibet. These are militaris. These grow in our environment readily, are abundant. So it's the ones from Tibet that are a black market or a trafficked thing. The militaris are here, and apparently some research showing that eventually the compounds that are desirable by these fungi can be produced equivocally by the militaris under certain growth conditions. So I don't know, be cool. Tibet's pretty high up there, but I would say just it's a cool pet project, but I would probably just do something that's the path of least resistance. But potentially, I don't know. This is why I love mycology, because it's such an wide open. It's like there's so much to be experimented and done. This is why I say, like, with mycology, like, experiment. Don't be afraid to experiment. So yeah, for sure. Potentially. So paleomycology. So this is really neat. I like history, like natural history, like old stuff. Why not old fungi? Cool. So let's talk about botanical taxonomy. Well, just taxonomy in general, but botanical refers to plants, but in botany. But yeah, anyway, so here are two diagrams. One's a little friendly, one's kind of scary, but we'll walk it through it. So on the left is a very nice, simple example of of explaining the three domains and 5 kingdoms of life. as we understand it to this moment. So it's great because you can hold out your hand. You can hold out your hand. So everyone's got a hand. Doesn't matter if you're left-handed or right-handed. You can just kind of work off the fingers you got. So imagine your thumb and your index finger pinched together. So that's those are prokaryotes. Those compose the two domains of bacteria and archaea. And then if we move on over to our palm, the upper part of our palm, our middle finger, our ring finger, and our pinky, that is the realm of eukaryota. That is the domain of eukaryota that has the kingdoms of plantae, fungi, and animalia. So it's really interesting because in general, like life started as these, single-celled organisms and bacteria and archaea, and then we understand that at one point, an archaea enveloped a bacteria, and rather than digesting it, it went through a process called endosymbiosis, where a mitochondria, which is pretty much an enslaved bacteria that produces ATP, and chloroplasts, plastids that perform photosynthesis, were encapsulated. With animals and eukaryotes, simpler and higher, the idea is that this was maintained and that you have a combination of symbiosis. So at what state did symbiosis become a direct interpretation? We talk about vertical transmission. How long does it take for vertical transmission to happen and propagate to a point where it's indistinguishable from the larger organism at play? That's kind of cool. Oh yeah, sorry. The enslaved bacteria one was a meme that I saw. It was one of those weird, like, it's like a amateur head thing where it's like, yes, enslaved bacteria, but it's in symbiosis. It's 1 + 1 = 1. It's not extracted. Everyone benefits, even though the mitochondria does exit the cell, the immune system does apparently attack it. Anyway, cool. So bacteria and archaea, endosymbiosis, mitochondria and plastids and plants, photosynthesis and cellular respiration. Woo! So we look at our hand, thumb, index finger, bacteria, archaea, a prokaryota. Over to our palm and our ring finger, we have plantae, and we have biconts. Plants are biconts. Then we have opisthocons, which are the range of protists, fungi, and animalia. So biconta versus uniconta. It's the concept that the ancestor had one flagellum versus two flagellum. So biconts had two flagellum, which turned into plants. And then with the unicons, they had one flagellum. And then we move on up. Biconts become plants. Plants are biconts, but in a large sense. And then you move on to your palm. You have opisthocanta, which are kind of these. Between there is protista. Then you move up to Holomycota, the kind of associative, fungi related to the aspect. Taxonomy gets kind of blurry between the fringes. And then the proper fungi, and then we have. Then we have animalia, which were holozoa. Woo, cool. So that was the fun and friendly one. And then we have the scarier one. So we've gone ahead and clipped it down just to uniconta. So we look at the aspect of uniconta. We have pisthocons and amoebozoa. Mind you, this is not a set in stone taxonomy. People are potentially always, like taxonomy is interesting because depending on who you talk to, you either be in agreement or get into a fist fight, because a lot of it is. We go off a concept of keys and how structures are formed in this kind of ontogeny or ontology, mispronounced in some way, some kind of malaprop. Anyway, but the reality in general is with DNA technologies and sequencing, we can actually do molecular clock analysis where we can basically compare certain key proteins, how conserved or how variable they are in their genome and their structure of their sequence of amino acids that eventually fold into a protein. So it's the kind of molecular clock. Anyway, so simple. Uniconta, amoebozoa, opisthocanta. Then we have holozoa, so we get animals and another Latin word I forgot. And then we have Holomycota. So Holomycota, we go up in kind of the nuclearids, the kind of interesting like slime molds. We don't really know how to place them. We have a lot of things where they're kind of like, they're just there. We don't really know where to put them. This is a working model in progress. And then we have fungi. So the fungi we've been mostly talking about this lecture have been the dikarya, which are the Ascomycota and Basidiomycota. Now, these are the ones that we're the most familiar with because they predominantly exist on land, right? Now people maybe say mushrooms are from outer space or this or that, or or pansporia instead of panspermia. Well, the reality is the fact is life has existed far less time on land than it has in the ocean, right? So if we move up to all those, we get a bit of the really weird, interesting fungi that, have been around for a while. They do a lot of different things, and we're only beginning to understand them. And like we talked about aquatic fungi, we're like, what? Not just like mushrooms that, there is a mushroom that actually can live underwater in streams and estuaries, but also the fungi, the actual fungi that don't produce fruiting bodies that exist in aquatic ecosystems. So pretty wild. Cool. This is a slide. I keep it just to talk about like keeping interpretations about science and how a lot of it is, if you read something, it's not fact, you got to interpret it. So this was a research vessel. Chiyuku is a kind of the equivalent of like the Geological Survey of Japan that would go around and basically look at rock sediment cores, which is pretty important if you're an island nation like Japan, which is very vulnerable to like a number of earthquakes and tsunamis, being right along the rim of the Ring of Fire. And so basically, in general, they went off to this geological formation northeast of that region of Japan, where you see the arrow basically placed in general, and that they basically drill into it. And it was a 20 million year old geological formation. They drilled it, and they basically think they found like this pocket of what was some kind of like mycelial mass or organic mass that had fossilized in some capacity. So apparently, they said they found spores in the sample, and they went to germinate it, and the mycelium grew, and then apparently fruited. And what fruited was in this petri dish that came off the petri dish in general. It looked an awful like Schizophilum communae. Schizophilum communae is probably one of the most, you'll see it everywhere all over the world. It's probably the most cosmopolitan fungus. It has an absurd amount of mating types for its genes. So meaning that, so we as humans, we have two types of gametes, meaning that they're complementary, meaning we can potentially biologically, numerically outbreed with 50% of the population at any given time, at any given moment. Given the fact that that Schizophilum community has such a large combination of loci and alleles, they could potentially outbreed with upwards of 98% of their population. So that you basically are allowed to recombine any circumstance. That's why you'll find them sometimes all over the world, but also where they're consumed or just casually in people that have developing or suppressed immune systems. They've been found in the nasal passages growing in children that have immune conditions, or even just ones that have developing immune systems. And they've even been sometimes found in polyps in the mouth of some older people in cultures where they actually do consume Schizophilum commutum, because this is a fungus that is able to survive in many conditions and then basically breed in a number of conditions with the huge range of mating types from the wide range of loci and alleles that are dedicated to that. That was 20 million years old, I think, but it's open interpretation. I think the core probably got like contaminated with a spore because it's the most common fungus. But what's the appetite for paleomycology? What if? Cool. So 99 million year old mid-Cretaceous agaricomycetes encased in amber from Myanmar. So I'm sure we've all seen Jurassic Park. We know a great way for understanding what life was like a long time ago, because organic matter doesn't fossilize well. All good. No worries, Catherine. All good. Fossilized well, it can be preserved in basically the stickier, higher sugar concentrate saps that basically copal that turn into amber in general. So we see this paracel-like mushroom, and we see actually an insect, some kind of trying to eat it as well. And what's really neat is we have this almost 100 million year old mushroom. Then we have this contemporary,
Erasmus,
which is amazing. So it's like pretty crazy that you have this morphological similarity existing for over 100 million years. And it's the fact is that, fungi, once you figure out a good form, keep it going. And there are definitely permutations and even convergent evolution where forms do come together and form again and again, independent of one another in general. But it's amazing to look at that snapshot, this echo of time compared to a contemporary fungi. 115 million year old Guanda caraketes magnificus from Crato Formation in Brazil is a tripe basin. So this was an interesting story. There was, I believe it was I forget where, it was probably somewhere Minnesota or Wisconsin. I really feel bad for mixing up the two. Anyway, but someone was looking at plates of these limestone, and they were looking for insects that potentially were recalcified. So organic matter doesn't fossilize well. That's why we tend to see bones. You know, insects and fungi, they're made of chitin, so they don't fossilize per se, but they can be calcified. So what happens is it's the thought that a mushroom fell into like a bog and landed in the silt and sediment, and then basically as the silt and sediment was fossilized into limestone, the organic matter was calcified, and you have this echo, this prepense. They were going through a bunch of plates where they were looking at insects, and they see this structure like, wait, that doesn't look, that's not an insect. It looks like awful like a lot of mushrooms. So they basically looked at it. They did a couple imaging screens on to look at the strape. They could say, okay, we can see what's thought to be a cap, the pileus, the gills, the stipe, and then also too, they went in and zoomed in with a scanner electron microscope to all the way down to a scale of 50 microns, and we see that these ridges, these potential gills of what would have potentially been a hymidium or a genetically rich area where potentially Basidia or some spore-producing surface might have been, but we can't see any spores. So another aspect of potential mycology 150 million years ago. So this is the Atlantic Basin in Brazil. So this is where it is geographically in reference to South America and the equator in Brazil. And then we see the actual location, and then we see the strata of the formation about how many millions of years ago. 400-million-year-old late Devonian prototaxis. That's prototaxis, though, what the guy is sitting right there in front of. I believe that's from Egypt. So prototaxis are really interesting because it like more or less means like the first ash or the first axpin for anyone who's really up to on their Latin for taxonomy. For basically sorting genus and species. And so when we were first, so when paleontology was looking into these aspects of these big structures, oh, these are huge fossilized trees, these are amazing looking structures. And so it was interesting because like upon further review, they realized these weren't like vasculatures like phloem and xylem, but potentially they were these interlacing matrices of basically what would have been in general, like hyphae and algae, that they might have been these primitive lichens. And so it's amazing. These things are huge. Like they're a meter by like 8 meters. That's like 3 feet wide by 26 feet high. Like it's tremendous. So with a little bit of it, so in general, we talk about, okay, what is a lichen, right? So a lichen is really neat because it's a symbiosis of a fungus and a photosynthesizing bacteria or an algae. And what happens is, in general, you have the fungus growing. So when you see those kind of leafy little lichens in your cemetery or in your parks in general, you have a fungus that's providing this protective kind of this protective meshing, this intertwining that is breaking down and digesting the substance it's growing on. Like we often see them a lot on wood, or even mostly in cemeteries or urban areas on concrete or some kind of stones, and digesting that, extracting the macronutrients and trace elements that are in that combination of mineral, and exchanging it for sugars from photosynthesis produced by the algae. So you have these algae suspended above, and then what's amazing in general, you have this fungus. It's not just one fungus, it's often a combination of funguses. So it's actually a tribont. So you have the symbionts. So the word symbiosis actually was invented to describe like lichen biology, but it's oftentimes cyanobacteria or algae, and often one or two types of fungi. And then based upon the type of fungi, it'll actually affect the macromorphology. So it's interesting, they can actually reproduce by spores, by the ascocarp, the actual spore-producing structure of the asci, but also too, these little sections of the hyphae and the algae can break off, called sporidia, and land on a surface and start propagating and start continuing this biological dynamic process, which is just incredible. So here's an artistic interpretation of what these might have looked like, towering over the Devonian landscape 400 million years ago, as most plants were only a meter tall. So pretty amazing and interesting to think about that. Let's see here. So this is a fun one. I think this is the most recent one that came out. They did a molecular identification of Neoproterozoic shale rock. So this was similar to I believe the one that was from the Crato Formation in the Atlantic Basin. Feel free to have a look at these papers. I'll probably put a paper list together and send it to people. But yeah, in short, they basically use a number of techniques: SEM, TEM, confocal microscopy, Raman spectroscopy, all these different details, and also molecular identification to basically look at the similar formation and assess that this sucker is a fungus that's a microfossil, and also it's dating back to roughly 810 to 715 million years ago. That's really old. And because like papers are kind of dry, here images, which are always a nice relief, and showing the scale and with microscopy, and you see the potential hyphal shells. Another one also I think from 2018 or 2019, they formally named it Orospira geraldae, early fungus from the protozoa there in Canada, roughly 1 billion years old. This wasn't extracted. It's more so a spore that was germinating encased in resin. So it's basically a biological process, like copal we talked about, that becomes amber when it's fossilized, or in general resin, which are sticky, that are, basically do well with time as things are perceived in general as well. So it's pretty amazing to look at that. And so, and then that's kind of the revised, taxonomic tree that is built out, and potentially with the dating to that. And then with respect to living fossils, we think about how, and we're talking about the fact that probably the first plants formed, the first life moved onto Earth in a combination with bacteria, cyanobacteria, algae, and fungi, making these primitive lichens, these protoplants. And we understand that the first soils, they were barren. They were just like rock, like sterile. Like there was microbial activity, but you had to have something to basically break down the microbes, which was like this proto-fungus. And then eventually, rather than decomposition, you have a symbiosis. We're like, hey, I can break things down, inorganic matter, organic matter. And in essence, I can pretty much make these symbiosis. So here's an existing fungus, Geosiphon piriformis, which actually does contemporarily make symbioses with I'm not sure if it's both, but with photosynthesizing microbes, algae, or cyanobacteria. And the reality is that based upon looking at its ribosomal DNA of the small subunit, the structural, using a molecular clock, is contemporary with dating back older ones. I can link the paper. It's technical, but it's an amazing concept of looking at a living fossil versus all these stacked ones in general. Fungus-like mycelial fossils in 2.4 billion year old vesicular basalt from Paleoproterozoic ongoloid formation in South Africa. So to make a claim that something is this old, you really have to like do a lot of like due diligence to like back it up. That's why the previous one with the DNA analysis from 715 million to 800 and change million, they did like upteen number of imaging and processing on it, because if you're going to make a claim, you better back that up. Like that's just how it works, but it's amazing to think about. We learn that much about our environment in so many different cross-disciplinary fields. So basically in general, we have a fungus-like mycelial fossil in 2.4 billion year old vesicular basalt from Paleoproterozoic ongoloid formation in South Africa. Okay, let's like put that into English, or like in language that does not put someone to sleep. So fungus-like mycelial fossil, like filamentous structures that seem like hyphae, that match like the shape and size. 2.4 billion year old, pretty old. Like it's basically Paleoproterozoic, so before the early stages of protozoa. And it's from this ongolong formation in South Africa. So the basalt is lava rock. Vesicular basalt is like one type of rock. This paper is really interesting because like the first 5 pages, like, am I reading a paper about fungi or geology? Like, which is cool because it shows the cross-disciplinary aspects of that, like helping people understand these details to make your argument better. So ongolong formation is a large formation that goes over, to my understanding, Southeast Africa. So they basically looked at this capacity where you have these calcifications, and you going down all the way down to a 50 micron assessment, which kind of fits the bill for a hyphae in that scale, 5 to 20, like that, anywhere between single digit to double digit hyphae of these potential filaments in general. Cool. So it is now 6:26. Damn, right on time. So cool. So this has been my presentation. I'm going to do questions and answers. Here's my contact info if you want to stay in touch and contact with me in general. And yeah, so unless y'all want to ask me any questions, let me know. If y'all did appreciate the presentation and work I put into this, I would appreciate if you could donate. You can donate through PayPal, which is available through my Instagram. You could also donate through LiberaPay as well, which is you can do recurring or you can do one time in general. And what I'll do is I'll just kind of pause the presentation and maybe kind of show you guys where to donate. So I hope you all enjoyed what I gave today to you. Whatever you feel is appropriate. You know, I'm not going to put a dollar. If you found this valuable and insightful, and also it helps me provide a taste for what could be provided in the future and more developed, I'm not going to ask for like a flat payment unless it's a fully brought out course. This is more so a compilation of the work that I've done, and I feel like with the current state our world is in, we need to have more of a communal, symbiotic, mutually supportive environment than a transactional one. So it's the way I've done business in the past, just being very collaborative, seeing we'll make it work in some way. So yeah, so you can support me. Go to my Instagram. It's Instagram.com MYC.NYC. Go to my link tree, this one right here, link tree, and you can either support me through PayPal, which is pretty self-explanatory. If you don't have a PayPal account, or if you're not in a country that supports PayPal, or you just would rather pay with a card, you can do LiberaPay, which is a Patreon alternative. LiberaPay is great because it's open source. It's not owned by a company. There's no fees associated. I get paid directly. It says recurring payments, but you can do a one-time payment. You just set the amount that you want to donate. You set a custom amount that you want to donate. So let's say hypothetically 25, hypothetically, you just do like a per year, and then basically you select manual. You would donate, and then you go into the profile, and then you basically would just cancel. After donating, you would just cancel the recurring thing, and it's pretty straightforward. And what's really cool about LiberaPay is that all the donations that I get are publicly displayed and available, and I plan to giving some back to the actual organization LiberaPay themselves, which they're self-work. So it's an open source payment platform and transactional platform as well. So on that note, this definitely has been an awesome time. I hope you all enjoyed it. Yeah, I'm gonna be uploading the recording. So I've been recording it the whole time. It takes about a couple hours to download it. I'm gonna go and edit it, so it's really effective for everyone as well. And I'll have some time this weekend. Yeah, YouTube. I have a YouTube for sure. I'll make it super accessible. And then also I'm planning to make a list of all the papers that I talked about today. You know, if you don't have institutional access to get a paper, you can often just email the person, and there are other ways, wink, wink, to get a paper. But as long as you have like the name of the paper and the authors, you can definitely get in contact with someone to articulate how a paper does as well. So I'm going to be sending up a follow-up email, probably with like the poll, like to get some feedback from y'all. I know I hate filling out polls. They're really annoying. Like they take 5 minutes, but like literally they take 2 weeks to do, just because like, you know, everyone's got a million other things going on. So in general, you know, I'll just do a follow-up. Also, I'd love to do some more classes in the future for y'all. I think some people express interest in the cardboard and coffee. It's a great way to make a passive system that can literally eat your waste, and then you can just put it in your garden. It's a great amendment. You know, really exercising a circular dynamic like there as well. So happy to do one. If I do a class, I want to make it so it's like defined content and really structured. This is more free form. This is more a collection of all the stuff I've done over the past 2 years, kind of mashed into one relaxed thing as well. But for classes, like if I'm asking for a set payment per admission, I want to give you something of value. So that's just how I personally do things, because I see my work as a reflection of myself, and also want people to benefit from that capacity. So this will be uploaded. I'm going to be sending a poll for some feedback. If you guys typed what you liked in the chat right now as well, that will be recorded, and I can kind of go through that as well. My contact info is on my gram. Oh, thank you, everyone. Sweet, for sure. Thank you for coming out. And I think, I don't know, I think the perspective that I do provide on fungi and mycology and how it's progressed more into beneficial microbiology and permaculture systems, because like I'm a very visual learner, and like so a lot of it is understanding the whole system and being able to articulate and visualize to people. I think there's a lot of potential for like how these connect into other spheres and fields with fungi. So I'm glad, I'm happy to share that, and really like to think about how we can look at our world from a permaculture perspective and look at our world in a science perspective. Like we have permaculture principles and first principles, logical versus much more feeling and kind of aspect. I always kind of see like a spectrum where you have a spectrum where on one end are people that are much more kind of the Rudolf Steiner biodynamic kind of ethereal kind of Jungian kind of thing, which is a bit more mystical. On the other end, you have the logical, like the bleeding edge of science, which is like synthetic biology. And oftentimes people in these camps want nothing to do one another, which is unfortunate because I feel like there's a lot to be learned from one another. This is a whole kind of corpus of human accumulation of knowledge, and while people have the best intentions, it's about context and understanding about what may actually have happened, what may have happened at one time. So I think about with technology, it's we tend to view the world and how we can articulate or manipulate or manipulate or control or refine our environment. But we think about biotechnology, right? We think about biology and how we interact with it. A plow that's used to break up a bit of soil to do some tilling or some low till or no till that our ancestors figured out during the start of the agricultural revolution is just as much a piece of biotechnology as a pipette. And we tend to forget that. I think a lot of it is connecting these ranges where we're synthetic biology, we're literally synthesizing new strands of DNA and incorporating those into selected model organisms to express molecular programs to produce things that are viable and important and could be revolutionary and done in a manner that's responsible and safe, and it follows certain strong ethical principles motivated for advancement rather than for profit. And on the other end, in general, we have people that are following examples of nature and biology and systems and observing things where there's natural patterns being observed, but they're so old and so strong and so maintained that it comes from places in human history where we lacked a vocabulary or a science to describe or articulate what's happening and kind of the resolution where we have this curiosity. And I feel like pulled in from each, like towards the center, we have permaculture on the end of kind of the more ethereal biological focus, more of the agriculture aspect of biotechnology, which is amazing principles about how do we mimic and emulate these conditions overall. And then on the other, we have molecular biology, which is becoming more accessible now as the price of sequencing and synthesizing DNA has become more and more affordable, that you could get this equipment together, and people are doing it to this aspect, like shown by the community biology movement. And I think there's a lot to be learned from either group immensely, and that we're kind of at this really incredible time in human history where we can integrate these aspects. And I think serendipitously a time where, our species and the fate of our planet, and even the civilization we resemble as what is contemporary today, remains. So wanted to thank you all for coming out today. I'm going to post a recording this. Again, last shill. If you did enjoy the work, please consider supporting me now or later, and I'll be posting up the information in the next coming days. And I think I have everyone's email list from the Eventbrite. So I will do a poll asking you if you'd be like to opt in or opt out of any future communications and just with feedback on the course. And if you would not like your email used, let me know. I'll remove it. I have the database. I think with any information that's collected, even from a system like Eventbrite, people don't realize it's being collected. I definitely want to make sure that I'm being transparent in how it's all being done. So I really believe in non-transactional, open, collaborative, and transparency in how I operate and do things as well. And so also making it open source. But yeah. Anyway, let me. Ooh, there's 57 things. So maybe we'll answer some questions. I just want to kind of go over real quick. So that before I did the wrap up spiel. Okay, cool. I'm muted. No, so there we go. Okay, let's see here. So almond mushrooms, like almond agarics, yeah, it grows all over lawns in the summertime. You know, they're super tasty. You know, I think I was upstate New York with some friends last summer, and we were driving, and you could actually spot them on the road. So we were driving on these back roads in rural areas where the soil is in pretty good conditions, and we would just stop and then pick mushrooms and leave. So yeah, they're great. Let's see here. So yeah, so I'd be down to do a webinar talking about DIY home cultivation techniques, and a lot of it is, now that everyone's kind of home, like a lot of what I've been doing now is like kind of absorbing all of the information I could. Like this is, I'm like some of the books I have. I don't know. I'm a big book hoarder. A lot of it is just like taking all the information I can and absorbing as much as I can in a timely fashion, if there's only enough hours a day, and kind of looking at the connections to like, okay, like what do I need? Do I have to follow these things, or can I experiment and really trying to peel them down? It's cool to do like for laboratory technique, like it's good to do a lot of like the sterile technique and down detail things, because if you want to get data and results, but if you just want to experiment and try, like that's the thing I love in general. Let's see here. Home remediation, that'd be more like preemptive stuff. There are some cool exercises that you can grow like mycelium on like on a rag soaked in bite grease from oyster mushrooms to kind of articulate those details. Oh, hey, Leaf. How's it going? Cool. Yeah, I'd love to stay in touch with you too. And love to stay in touch with anyone. Cool. 2-3 hours. 2-3. Cool. Yeah, Adrian, feel free to shoot me a message. Cool. All right. Well, it is now 6:41 Eastern time. Oh, how does citizen science get funding? I kind of just go for broke just because I'm insane, because I just teach and keep propagating. I'm getting better in the grant funding thing. There's a lot of good resources for grant funding. If you two really good organizations that merge into one, it's GuideStar and what was it? Foundation Center merged into one. Now they're usually provide resources finding grants for not-for-profits. If you're interested in doing a citizen science, I think one of the best ways is to find a community environmental conservation organization in your area that has 501c3 status. And if you want to do work related to it and do good science, and maybe get paid a little bit in addition to like materials, resources, fiscal sponsorship is a real thing. It's something I've kind of looked into because, you know, it's great to do science and eat as well, you know, and also pay rent. And I think this whole situation is really kind of shifted a big reorienting in our concept about how we should relate and interact with nature and what that means on many different scales. But yeah. Cool. All right. Well, I hope everyone is having is having a wonderful day. I hope everyone is staying safe, staying healthy. And I really hope that going into the future, we can understand that our interaction with nature very much has a ripple effect. And I think about much like the way our activities and interactions need to be held responsible and how we work together overall. So yeah. All right. Thank you. Have a wonderful evening, day, or morning, wherever you are. And we'll be in touch.